a woman dies in India due to cervical cancer. But again, the good news is that get vaccinated and be safe. Simple as that. There are vaccines available for women between the age 9 to 45 years. The best age is 9 to 16, or in some cases 9 to 25 years. So I guess most of you are in this range. If you have not got vaccinated, please consult any government hospital or private hospital or a gynecologist and get vaccinated. It is not very expensive to do. And I think it's maybe a cost of one party, what you do, or a birthday party, and get yourself a good present for life so that you don't get cervical cancer in your lifetime. And I would request you to spread this message to whoever you come in contact with, to your sisters, in future daughters, or your friends, to get vaccinated and be safe. Okay. This is one of my favorite topic to talk about, stress, mental health, and cancer. So sometime back, I use Google or any AI tool to check whether stress is related to cancer. Can stress cause cancer? And the answer which it gave me were, these are the key studies. And it's difficult to read, so I've summarized it for you that most of the time it's no, or it says yes, but with a maybe or somehow. But there is no clear answer that yes, stress can cause cancer. Now, I dwelled upon this topic a lot in the last 6 months to 8 months. And then I finally came to the conclusion with this picture. So I'm sure you all have seen Three Idiots. And when he says that ये engineers बड़े चालाक होते हैं सर, ये ऐसा कोई device नहीं बनाते जो stress को measure कर सके, right? And again, in continuation with that, there is no publication, no in literature available which says that stress can directly cause cancer. Okay? But I'll prove you with my knowledge and with my understanding that how stress can cause cancer. And I assume that after the end of this slide, you will agree with me. So when we are stressed, what do we do? Or what do normally people would do? Some people start smoking, right? And then it would make a habit because again, the stress never goes out. It only increases with your age, with your position, with your money, or etc. in various conditions. The next, some people would start drinking, even if they are drinking casually, very rarely these days. But again, with more stress, they would make it a habit that, okay, it would relieve their stress. So let's drink. The third would be, which most of us do, or the youth definitely does, is stress eating. So we try to munch anything what we have, or sugary things, because that would relieve us with stress. Or chocolates. That is also a very go-to thing that, okay, jab chocolate kha lete hain. And I don't know how many of you know that there is actually a loop with the biology that why you eat chocolates and why it gets you release of stress. So there is a chemical which goes to the brain and which it's a loop that you have to eat more and more sugar and chocolate to get rid of stress. Okay, please find the article. So this is your homework maybe to do, and you will find interesting to read about. Now, a lot of times this question comes about that, okay, not only these things, but does it come overnight? No, it takes years actually to develop cancer in your body. So when you continue those things for a long period of time, plus the environment where we're living is polluted, we are not having the best of the food. Even if you eat good vegetables, fruits, they are also contaminated with some chemicals or pesticides, right? So there are a lot of things which accumulate and which will lead to cancer or any other disease in future. Now, whenever I present this slide, there are young students or people who are asking this question that are these the only things which may cause cancer? Because you are highlighting those things that, okay, smoking, drinking, and junk food. No, it's not like that. So if you look in the publications, so 90 to 95% times the cancer is caused by environment issues, and 5 to 10% of the times it's genetic. Now, if you again subside to the 95%, it would come around 30 to 35% times you have to blame the diet, what you are eating. 10 to 15% times it's the other category, 4 to 6% alcohol, obesity, infections, and tobacco. And whenever I present this slide, there is a question from the youth कि अरे alcohol तो 4 से 6% ही है. Let's start that. कि अभी ये भी पिएंगे तो भी कुछ नहीं होगा. But again, it's not like that. It comes with a lot of additional bad habits, and then it will increase the chances of those diseases. Now, this is a very basic slide which mentions about what are the treatments and why is it so difficult to treat cancer. So I'm sure you all have heard about these treatments, which is provided to a cancer patient. But why is it so difficult to treat? Maybe this is new for me, or this is new for you guys, and little interesting also. So it starts with one mutation, and then with time, there are multiple mutations which are acquired in the body. And then someday there is a driver mutation which actually blows up, and the cancer is visible or known to the person and the doctor, right? And then you get the treatment, and again there is a slump, but there are still some cancer cells which are still remaining in the patient. Also, the therapies what are available these days, chemotherapy, radiation, they are also mutation causing, and they are also cancer causing agents. So again, there will be multiple mutations with the remaining cells, and the cancer comes as a relapse. So this is the newly clonal evolution model of cancer. Now, are we doing all right with our healthcare system, or is all well? However we are treating patients or handling them? So this again may surprise you that average time spent by a doctor with a patient country-wise. So in Southeast Asia, which is India, Pakistan, Bangladesh, China, etc. So in India, it's less than 2 minutes. So just assume that if there is a cancer patient, you go to a doctor and he spends 2 minutes with you, or with, not you is a wrong term because I don't want anyone of you to get infected or get any disease. But anyone who goes, it's 2 minutes is like nothing. I always give this example that let's say कि कोई गया और वो डॉक्टर बोल रहा है कि हाँ डॉक्टर सक्सेना, सॉरी आपको स्टमक कैंसर है और आपको 4 राउंड ऑफ कीमोथेरेपी लगेंगे, 6 राउंड ऑफ रेडियोथेरेपी लगेंगे. द मोमेंट दैट पर्सन हर्ड कैंसर, वो स्विच ऑफ हो चुका है. ही इज थिंकिंग व्हाट विल हैपन टू माय वाइफ, व्हाट विल हैपन टू माय ब्रदर, सिस्टर, daughter, or my parents, because he thinks that is the end of life, right? Because हम डरे हुए हैं कैंसर बीमारी से. Yes. Time? Yeah, I'll rush up. So there is a need for palliative care. Basically, doctors are too busy. There are diets, lifestyle plans, which are important in cancer treatment, post-treatment care, and also the psychological help for the patient. And this graph shows that if you take palliative care with the patient, they survive better. So this is the work what we are doing. So the organization name, I'm the founder of the Dr. Acha. So it's an acronym ACHHA, which spells A Cancer Health and Happiness Advisor. So we are trying to create awareness with people, answer all the questions of the cancer patients, psychological help, diet management, etc., etc. And you are welcome to be ambassador of this work. And today is the World Mental Health Day. So we have started our new initiative, Happiness for Health. We deal with mental wellness of youth, personalized methods, and help. So again, I am happy to welcome if there are any students or anyone who is happy to help us as a volunteer to create awareness about cancer health or mental health. Please contact us or join us. And last slide, don't trust technology for your health and well-being. As I said, I use AI a lot. And then there was an article recently in the newspaper that many Indians are treating AI as their doctor and turning critically ill. And I asked this question to ChatGPT that can you replace a doctor? Is it possible? And the answer was no, I cannot. So please talk to an expert. If you have any questions, that is always better. You can also type this question and ask ChatGPT. Okay, so where are your 1 to 2 pros? So if you stay healthy, you don't have to spend that much money for any treatment, and you will save that money. With that money, you can travel the world, do whatever, buy Discovery, a Land Rover, whatever, and enjoy, rather being into the mess of healthcare system. So thank you, and wish you all stay happy. Questions, if any? No?
So family is having question. Okay. So, sir, I have some question. So how we can reduce the risk of cancer in Indian population? I just say this cancer and other lifestyle disorders on rise in India compared to other countries. Right, because in junk foods and all these things are being used in other countries as well. Why such trends are seen in India and how we can reduce them?
It's a very good question. So if you talk about junk food in US, so it's long, they've been using long, and that is why their most of the population is obese. They also have cancer occurrences probably at equal rates what India has. Okay. But the thing is that the population or the genetic diversity what US or Europe has, they are not based on that. Our genetically biota or the microbiome what we have in the gut system is very different from those. So every youngsters they always ask this question, sir, कि यहाँ क्यों पिज्जा और पास्ता को बना दिया जाता है? Because in Europe or that is a staple food. Because their gut microbiota is actually worth or using basically that kind of cheese or India में कभी cheese hard cheese produce होता ही नहीं. It was paneer it came in 1800 or so. We are not used to those things. We don't have the microbiome for that. So this is one of the reasons that geographically we have to eat what is grown here, what is locally cultivated things. For example, like one simple the example like which I heard or I think read that don't eat anything which doesn't have a Rajasthani or an Indian. For example, I'm starting, right? अभी आजकल हर कोई इंसान है उससे ही सारा न्यूट्रिशन मिलना है उससे ही सारे डिसीजन खत्म हो जाने ऐसा नहीं है आपको वो चीज खानी है जो लोकली इंडिया में अवेलेबल है जो यहाँ मिट्टी में यहाँ उगती and not getting from Mexico and eating it. That would help you.
It's a very insightful presentation, and we are very thankful for you to come here and enlighten our students. Thank you so much.
Thank you, Dr. Amit Sharma, sir, for an enlightening and holistic perspective connecting science with wellness.
To express our heartfelt gratitude, we would now like to present a memento to our respected guests as a symbol of remembrance and appreciation. I request Dr. Gaurav Kumar and Dr. Ravi Ranjan to present the memento to Dr. Amit Sharma.
Our next invited speaker is Dr. Sanjeev Kumar from Guru Ghazdas Vishwavidyalaya, Bilaspur. They will be here with us virtually presenting on pharmacophore-based high-throughput screening, molecular docking, molecular dynamics, and MMGBSA approach for identification of prospective UDP 2,3 diacylglucosamine hydrolase inhibitor from natural product database against Salmonella type. Sanjeev Kumar is an accomplished academic currently serving as an associate professor at Guru Ghasidas University with a distinguished record of teaching and research in molecular biology and bioinformatics. Previously, he held the position of associate professor as well as assistant professor at Vellore Institute of Technology, Center for Bioseparation Technology (CBST), where his research focused on structure-based drug design, monoclonal antibody development against antimicrobial resistance, and lipid A biosynthesis process. Sanjeev Kumar also contributed as PhD Young Scientist at NIT Rourkela and held earlier academic roles at Central University Bilaspur and All India Institute of Medical Sciences. His expertise spans academic advertising, university lecturing, computer-aided design (CAD), higher education teaching, and educational technology, making him a versatile educator and researcher with a broad, impactful career in life sciences and bioengineering education.
We welcome Dr. Senthil Kumar to deliver his talk.
Yes. May I audible? May I audible?
Yes, sir.
Yes. Okay. Slide is visible, student? Slide visible or not? Slide visible?
Yes, sir.
Slide visible. Okay. Okay. So thank you for a nice introduction. First of all, I am thankful to Dr. Sanket Kaushik, who has invited me for this conference. And I am extremely sorry I could not. It is visible. It is audible. I am audible.
Still is not visible.
Slides not visible. 1 minute. 1 minute.
Just 1 minute. Right.
Sir, at your end, your slides are visible. I think there is some projector issue. That's why they are not able to see.
1 minute.
Sir, your slides are visual, sir, but their projector issue, so they are not able to see.
Okay.
They are rectifying, sir.
Okay. Okay.
Sanjeev sir, really sorry for the delay, but sir, we are facing some technical issue.
No issue. No issue. No issue. No issue. No issue.
हमें सिक्का वाला का हाँ क्या तुम्हें नहीं करना ठीक है मैंने कंफर्म नहीं करा लेकिन तुम्हें नहीं करना पूछेंगे भी ठीक है
कोई भी मतलब तो जैसे उसे रहने की इसके वन अनुसंधान से क्लियर दिक्कत आ रही तो जैसे उसे रहने में वो तो सर दिक्कत आ रही प्रोजेक्ट
फ्लैश पर पूरे में रवि सर के साथ पढ़ लिखो ठीक है बेटा सर को बोलो कि
Sorry for the delay, sir. You can continue with the presentation.
No issue. No issue.
Sandeep sir, sorry to disturb you. Sir, your slides are not in the slideshow mode, sir.
Slideshow mode. Okay, 1 minute.
Yes, sir.
1 minute. Now it is visible.
So the slides are visible, but they are not in the slideshow mode.
Okay, 1 minute.
Sir, you have to stop share and then again share. And when you share
Okay.
it, then sir, just choose the screen option. Because
Okay.
your office version is old, so that's why
Okay.
it's not coming on a full screen.
Now it is okay, fine.
So just
So you
put in
have
a
to make-
presentation mode. Put in presentation mode, sir.
Now fine.
Yeah, it's perfect.
Yes, sir, it's perfectly fine. Thank you, sir.
Okay. So
And sir,
thank-
your camera is off, so just on.
yes. Is it fine?
Yes, sir.
Yeah. Thanks for nice introduction. And I'm thankful to Dr. Sanket Kaushik, who has invited me for the talk. And I'm extremely sorry I could not come physically. And I'm also thankful to Ida Upadhyay also. She is always here, updating the talk time and all for that also. And it is good. Dr. Amit Sharma sir has given a very nice talk. Actual, I enjoyed that his talk. And basically, I'm a structure biologist, and I was in DST Center, Department of Science and Technology funded center in VIT Vellore. My lab doing the structure biology. We are working on a diagnostic, vitamin D diagnostic, and developing the monoclonal antibody. And also, as my training is on structure biology, so we are doing the molecular biology and three-dimensional structure determination. And bioinformatics all are just a small part of our work. We use bioinformatics tools for screening the library and all. So today, topic is that pharmacophore based high throughput screening. Basically, drug designing are four method. One is QSR, quantitative structure activity relationship, virtual screening, repurposing of drugs, and pharmacophore modeling. So my lab is involving antimicrobial resistant, and we are funded with the ICMR for ICMR grant for this antimicrobial resistant to work. So just this slide, in 1941, a police officer died due to the bacterial infection. Not enough penicillin was produced, but in 2016, a female in London, actually, she has died due to the bacterial infection, and none of the antibiotic was working. Situation is in 1941 there was not enough penicillin. In 2016 we are producing so many antibiotics, and we unable to save that lady. Actual reason is that there is a resistant actual, and this is a serious issue. And as I came to know that most of the students are BTech or MSc background, so they know very well, because I'm biophysics person, they know very well what is antibiotic. And just I'm explaining things actual. So antibiotics, according to the World Health Organization, antibiotics are type of compounds used to treat bacterial infection. So we all know all these things actual. And but problem is with antibiotics is that suppose we all are taking the antibiotics, and we are trying to finish the dose also whatever doctor has prescribed. And if we unable to finish the dose, we don't have a guideline that where we have to return back the antibiotics. Generally what we can do, we just dump into the dustbin, or and that will carry to the environment and all. And one more thing, we could not understand, many people not understand what is antimicrobials and what is the difference between antimicrobials and antibiotics. If any infection happened, people used to take the antibiotics. But we have to understand that antibiotic is specifically refer to the drugs that treat bacterial infection. And that antibiotic is not for viruses, not for fungi, not for parasite. And when discovery of our lifesaver pre-antibiotics era, when that antibiotics has discovered, and from that time only that increasing in antibiotic resistant, and same year when the antibiotic has discovered, same year there is a scientists observe resistant against that antibiotics. And we were relaxed in 1980 that we got a like a magic bullet, and no need to invest the money on that development of new antibiotic development. So, and what is AMR? Ability of microbes to evolve genetically to resist the effect of antimicrobial drugs. We all know that what is antibiotic resistant and what is the reason: over prescribing antibiotics, patient not finishing their treatment, self-medication, overuse of antibiotics, and poor infection control in the clinics and hospital. And it is really great that Amity Jaipur has chosen this topic, emerging strategy and innovation for the combating disease in animals and plants. We are agriculture-based country, and we depends on agriculture and dairy industry and all. And we are using antibiotics in case of animal, up to the 80% antibiotic used in animals are as a growth factor. And antibiotic prescription, 30 to 50% of the antibiotic prescriptions are unnecessary. If you have a suffering from viral infection also, we are by default we are taking the antibiotics, and that antibiotics cannot kill the virus actual, but we are taking. Same things with the animals also. 80% there is a no much guidelines. Most suffer is animals due to the antibiotics. Humans suffer less compared to the animals. Animal suffers more compared to the human. And death attribute and antibacterial resistant silent. It is a worse than COVID actual. What we have seen in COVID, and that is COVID has gone, and but this is the major problem. And just you imagine 2050 in Africa, more than yearly 4 lakhs death can be predicted, and in Asia that is more than 47 lakhs death can be possible due to the antibiotic resistance. And there is a loss of lot of resource and all due to the treatment of this type of things. My lab is working on Salmonella typhi, and we are searching for different pathway. And we all know there is no need for any introduction for type Salmonella typhi, but problem with typhoid, we are notice a lot of resistant against this Salmonella typhi. Estimated 21 million cases annually according to the WHO report, and concentrated in South Asia and other is more reporting the resistant against this Salmonella typhi. So that I have just mentioned India, Pakistan, Zimbabwe, 3 just from the Center for Disease and Dynamics Economic Policy. From that their paper I have took this data. But what is the solution actual? What is the solution for this? Solution is that we have to find out the new target. We have to find out the new target. So that what are the target, like a gram-positive bacteria and gram-negative bacteria, they have like a gram-positive having a peptidoglycan layer. So my lab is working on gram-negative bacteria, gram-negative bacteria of Salmonella typhi, And as a MSc student and BTech student, we know that in gram-negative bacteria contains the LPS lipopolysaccharide, and we know that in case of gram-negative bacteria, there is a lipid A layer is there actual, and this is lipid A layer where LPS is sitting actual. Lipid A, this is blue color where the LPS is sitting there. If you disturb the lipid A, then you will disturb the sitting place of the LPS. So that lipid A is formed by, this is our target, and there are so many companies and there are so many lab in US also, they are targeting the enzymes involved in the lipid A biosynthesis pathway. And this is our target to know the structure of various enzymes and develop the small inhibitor. RADS pathway. RADS is the scientist who has discovered this pathway, and this pathway having a almost 9 enzyme involved: LPXA, LPXC, LPXD, and LPXH. And among this LPXC and LPXH is rate limiting step for the synthesis of lipid A. My group is working on LPXA also, LPXC also, LPXD also, and LPXH. One my student, she has recently submitted the thesis on LPXH. So that we are working on LPXH. Till now, there is only one inhibitor is developed against the LPXH, that is developed by AstraZeneca, and that is also recently it has a lot of side effect and a lot of clinically failed, and so that lot of people are working on LPXH, and we have also started this LPXH work. And because if you disturb the LPXH, then bacteria will not synthesize lipid A, and lipid A will not synthesize, then LPS will not the place to sit, and it will a virulence factor of the bacteria will be totally decreased actual. In this hope, we started this project. And this is the only enzyme, only inhibitor, sulfonyl piperazine is the only inhibitor that is developed by AstraZeneca. And we thought what we have to do, we thought we have to do the expression purification of this protein, and then identify the small molecules that selectively bind to the module the activity of LPS-Act, either using Schrodinger and all, at testing this best hits against the various gram-negative bacteria. And or if you will not get the small inhibitor, then we can use the pharmacophore modeling and QSR to find out the small inhibitor. So what is the workflow? Just if you are MSc student, BTech student, you know you have already studied the molecular biology. We have isolated the plasmid isolation, then generally we synthesize this clone from the company, and we only express this protein. And this protein is a membrane protein, and we try to express in E. coli, and we found that this protein is with this type of work issue with the solubilization. And when I'm not discussing much technical things, just for a student, and we overexpress this LPXH protein, and we found that it is coming in the inclusion body that is insoluble. If protein is insoluble, then you cannot do any work. Enzyme, it is enzyme, you cannot do the any enzyme kinetics, and you cannot do the enzyme inhibition studies. So we have tried various method to solubilize this protein. You have already studied in your protein folding and all. We failed actual 1.5 year. We failed to solubilize this protein. We use the protein solubilization on column refolding. That also it is coming in aggregation, and it is coming in aggregation. Then we got from the Takara chemical and molecular chaperones we got, and you know that in chaperone we'll slow down the protein folding process, and by using this we got this protein in a soluble fraction. Just as a story I am telling to you people, a student, And then we purify this protein by using the IMAC, immobilized metal affinity chromatography. And this protein has been, then there is some impurities. So we use ion exchange chromatography and we have purified this protein and started the enzyme kinetics and enzyme studies and all the things actual. Later we will discuss in last slide about all these things actual. So then what we are finding out the lead molecules against these enzymes. So we have follow, because this three-dimensional structure of this protein is not available. We started the crystallization and still we are doing the crystallization experiment. So we thought, why not we have used AlphaFold, either Swiss model, iTester, MOE, Yeshara, and we can develop the homology modeling actually. People know that if the sequence similarities is like this LPXH from pneumonia, this structure is known, and pneumonia and Salmonella typhi having a sequence similarity is 80%. So we thought, why not we can develop the homology modeling. And before homology modeling, we try to understand the this active site and how what is the environment of active site and all. And substrate is lipid X. So lipid X is hydrophobic in nature. It will bind to the active site of the LPXH. So we just from that we can know that maybe that active site will be the hydrophobic in nature. Just one idea, rough idea. And from that we have developed the homology modeling of the Salmonella typhi. And we have used various software, Yeshara, AlphaFold, and Swiss model, and we found best model that will satisfy the Ramachandran plot and have this model. This is the model, and we have verified by the Ramachandran plot. And this is the cap domain where these are binding. And manganese is the metal which is important for the binding and activity of these enzymes. We have done the enzyme kinetics also in presence of manganese, and we found that it is without it will be some inhibitor. If you bind with the manganese, this enzyme is inactive.
Sorry to
So,
interrupt.
this is
Network is fluctuating.
what?
Say your network is fluctuating.
Oh, network is that. Okay. Now it is. I'm just sitting. Now it is fine. Okay.
Yes, sir.
So we have a Schrodinger software. So this is the software where we can, if you know the active site, you make a grid around the active site and you can screen the natural product libraries. We have some paid libraries and all. That is approx 7 lakhs 7 lakhs natural product library. And we use the filter among them. Filter means that we follow the Lipinski rule and other various parameters so that it will narrow down the screening system. And then we do the virtual screening, and whatever the best binding score from 7 lakh, I am not much discussing about, from 7 lakhs we got two molecules, that is 1914 and 38352, and AZ1 is the known inhibitor that is developed by the AstraZeneca. And we found 1914 and 38352 showing the binding affinity approx -12 and -3. And when we have done the MMGBS, also it is high. And we found that most of the active site molecules where AZ1 is binding, where 19114 and 38352 also binding, and we follow ADME properties and all, and we have finalized 19114, 1914 and 38352 is the best molecules for the bind to the LPX site. And these are the ADME properties. We have checked the log P value and it follow the Lipinski rule. Both the molecules follow the Lipinski rule. And what we found, these two molecules is the good for the inhibition of LPX site. What is the next we have to do? We have to do the enzyme kinetics, enzyme inhibition with these enzymes, and also we'll do the antibacterial activity. So we did the enzyme inhibition studies, but still we don't have a very clear cut data actual. So that what we have did actual, we have one before that we have done the molecular dynamic simulation. Simulation means that we various up to 100 nanosecond, we make a box, and then if it will be sustained to 100 nanosecond or not. So if you see that 19114 and 19114, all these three molecules are sustained up to the 100 nanosecond, and there is a fluctuation. If it will be fluctuation, that we remove that molecules. Here 39226 is fluctuating, so we remove all these things. We have to which is not fluctuating, and we found that 1914 and 38352, and we have started data 0 nanosecond to 25 nanosecond, then 50 nanosecond and 75 nanosecond and 100 nanosecond. We found these both the molecules are stable in the active site. Okay, fine. Then what is the next? What we have to do actual? So what we have did, protein ligand interaction we have studied, and we found all the molecules, all the 1914 is interacting with the active site molecule, like arginine is the active site. Same thing if you see here also, 38352 also binding to the active site. And we found that throughout the simulation, these two molecules are stable. Then what we have did, we ordered these molecules. Along with this, we have what we have did, we have ordered 19114, 38352, 38372 and 39226. All the four molecules we have ordered from the company, and we have did the enzyme inhibition studies, and also we have did the antimicrobial activity, and we found that 1914 and 38352 is showing inhibition against S. typhi, E. coli, and pneumonia, and P. aeruginosa. So all the two molecules is showing better inhibition against the S. typhi, E. coli.
Sir, sorry to interrupt you. It's almost time. We are running a little late.
Oh,
So, yeah. Can you please?
Okay. Just 5 minutes, I will finish. We found these two molecules, and then what we have did, we have a MOE, molecular environment software. So they have a pharmacophore we got a two molecules assay, and we found this two molecules is the best one. Then can we get the better molecules by using the pharmacophore modeling? Because we have a all the what is okay based pharmacophore modeling, and one is structure based pharmacophore modeling. If any enzymes having an inhibitor, non-inhibitor, from that you can develop a ligand based pharmacophore modeling. And if you have a protein structure, then you can make a structure based pharmacophore modeling. As we don't have a structure of this protein, this enzyme, so we prefer the ligand based report. Pharmacophore modeling model, in that model we can pass the molecules. Here we have used 8 lakh compound, and this is the model actual. This is the model, and if it will be fit in that model, that will be that will interact and that will give you the binding energy and also interacting residues 1615, 1553. All these 2 molecules, all from 8 lakhs, we got the 5 molecules. There is a time is limits, and just I will coming. And then we have checked the ADME properties, and we found that 1615 and 1553 is the best molecules. And then this is a normal, you have to calculate the DFT analysis. I'm not discussing these things. And then this all the molecules, we have done the molecular dimension simulation. If it will be sustained, then your graph will be constant, is like a blue in color. That's if you see 16115 and 1553, both are coming in a straight line. It means that your molecules is interacting with the active site, and this is sustained actual during the whole the simulation. And just we have overlap the 1615 with the lipid X. Lipid X is the substrate actual. Lipid X is the substrate. This is the molecules. You can see that yellow color one is our inhibitor and blue color one is the substrate. If you see that our inhibitor is binding to the in just if you superimpose, it will where lipid X is binding, this 16115 is binding. There is a room actually, you can design better molecules also, so that many companies like AstraZeneca and all are developing. By this side we are developing in below side one, if you see this thing, and we have 1615 and 153 molecules. And okay, fine, I will go to here. We found that in case of our molecule 1615, there are some additional interaction we are getting actual. Generally, arginine 80 is not interacting with the with the AstraZeneca compound, but our compound is interacting with the arginine 80, which are critical residues for the binding, and all rest of the molecule, rest of the amino acid which interact with the AZ1, same type of amino acids are interacting with our known molecules also, our new molecules also. So that we found that in contrast, the binding pattern of complex 1553 differ from both AZ1 and lead compound 1615. Specifically, serine 160 and arginine 80 is interacting actual in case of 1615. So okay, I'm just finishing here. I'm not discussing protein ligand fingerprinting. That is also the new technique to develop the small molecules. Okay, I think I will come to conclusion. So conclusion, we have got by virtual screening 1 molecules 1914 by 16115, and by MOE also we got the 2 molecules. Conclusion is that antibiotic resistance is the newest silent killer, making existing treatment ineffective. Therapy highlighted the need for novel antimicrobial. A combined computational approach involve virtual scanning, pharmacophore modeling, can develop the small molecules and better molecules. I am not saying the computational biology you can develop the new molecules by using the computational biology. This will only narrow down the state, will get the molecules, and you have to do the enzyme kinetic, cell culture studies, everything you have to do. It will only the pharmacophore modeling, virtual screening, QSR only narrow down the compound actual. From 8 lakh compound, we will come to the 4 molecules and 5 molecules. Okay. So I am acknowledged Rajsekhar Reddy, Kali Kishore Reddy, and they are in Rajsekhar Reddy is involved in synthesis part of the small molecules, And this work, Gromia sir is also from IIT Chennai. He is always my student, DC member, so he helping for the interaction and all. And this work is funded by Indian Council of Medical Research, and I'm thankful to NRF Pierre also, and that I'm also thankful to Amity Jaipur for giving the chance to just discuss about the AMR. So thank you. So any question?
So if anyone is having any question, we can ask. Thank you, sir, for this very nice presentation on emphasizing upon repurposing of drug and speeding up the drug discovery process by using virtual screening and bioinformatic techniques. Thank you so much for your presentation and accepting our invitation to deliver a talk to this conference. Thank you.
Okay. Thank you. Thank you.
Thank you, Dr. Sanjit Kumar, for your deep insights into computational drug discovery.
Our next invited talk is by Dr. Sarika Chaudhary from Bennett University. They will be here with us virtually and sharing their knowledge on regulation of KLF transcription factors in ovarian cancer.
Sarika Chaudhary is a faculty member at Bennett University with an extensive background in membrane protein research and advanced cell culture systems. Prior to her current academic appointment, she worked as a Ramanujan fellow, focusing on membrane protein structural biology and held research associates positions at the Council of Scientific and Industrial Research and other leading institutions. Her expertise centers on membrane protein expression in HEK, insects, and E. coli cells, as well as protein purification, crystallization, and structural analysis. She has demonstrated significant proficiency in diverse laboratory techniques such as Western protein chemistry, and stem culture, and advanced molecular biology assays. Sarika research contributions reflects a strong foundation in protein biochemistry, cell culture, and biophysical characterization, making her a skilled scientist and educator in the field of molecular biosciences.
I would request ma'am, please grace us with your knowledge.
Good morning, everyone. Can you hear me?
Yes, ma'am.
Is my slides visible?
Yes, ma'am.
Okay, thank you. So I would like to thank everyone especially the organizers for this opportunity to present our work here. And at the same time, I also apologize for not being present in person due to the ongoing midterm examination at Bennett University. So our lab at Bennett University is basically focused in 3 different domains. One is the cancer biology, tuberculosis, as well as the biotherapeutic production. So today I'm going to give a brief overview of our ongoing work in the field of the cancer biology towards the KLF. So here we are trying to investigate the role of the KLF, which are the Kruppel-like factors that are involved in the cancer. So KLF, they are having the broad range of expression among the different tissues, be it, let me just take my pointer. Can you see the pointer also? So the KLF, they are present across, their expression is among the several tissues, be it the liver, heart, or the lungs, or the skeletal muscles. So what are they doing? That is the major question. So here they play the key role towards the regulation of all different aspects, including the cell development, cell proliferation, differentiation, and apoptosis. So there are 16 KLF isoforms are found, and these 16 are divided into the 3 different groups. The group 1 has the 3 members, KLF 3, 8, and 12, and the characteristic of this group is that it contains the CTBP binding motif. The group 2 interacts with the acetyl transferases, and the group 3 contains the SYN3A interaction domain. Our research focus is towards currently the group 2 members. If we take a look at the schematic domains of the KLF, Here we can see that it has the activation domain as well as the repression domain. And being a transcription factor as the characteristic of being having the zinc finger domain. So there are the 3 zinc finger domains that are present. Like I mentioned that KLF, they are the key player in the cancer and they're involved in metastasis, being it the EMT, ECM, change, invasion, and as well as the angiopoiesis. So we can see some of them, like the KLF6, here the expression is inhibited, while KLF6 SV1, here it is, by interacting with the twist one, it is involved in the EMT. So our group, we started to focus on the KLF6 in the beginning, and KLF6, like I mentioned earlier, they can be the characteristic of the Krupel-like factor is they can be the suppressor as well as the activator of the tumor. So KLF6 itself is the tumor suppressor. There the very well documented literature is there that it acts as a suppressor. So it has the 3 different splice variants. I hope the students, they understand the splice variants. So there are the 3 of them. If we look at the over here, it is the KLF6 wild type. Then we have the KLF6 SV1 variant 1, 2, and 3. So the SV1 is the one that has the antagonistic effect. That means it supports the tumor. It is oncogenic. So now our lab got interested in this because how the KLF6 being a tumor suppressor and has the splice variant playing a totally antagonistic effect and being the oncogenic by its nature, while the other variant it is showing the same profile as the KLF6 and is the tumor suppressor. For the other variant SV3, the functions are still unknown. If we see here, so KLF6 activation means suppression, tumor suppressor. And if we inhibit the KLF6 SV1, which was the oncogenic, so again the tumor suppressor will lead to the tumor suppressor. So this study has already been published, and they are both of these activities, they lead to the cell cycle arrest, apoptosis, decreased angiogenesis, and the decreased invasion. As most of the audience here, I believe, is the MSc students or the BTech students, so they might understand what is the post-translational modification, also known as the PTM. So the PTM, as we all are aware, that they modulate the transcription. In case of the KLF also, they modulate the transcription by interaction with the binding partner. It has been observed in the KLF that they have the variable impact, and they are known to be the molecular switch. So that means KLF, whether they are being the tumor suppressor or the activator, this role is employed by this molecular switch, which is the PTM. Hence, our interest came into this that, okay, we understand that they do control the subcellular localization, interaction with the other protein, and the transcriptional activity. So to understand this, when we were going through the literature, we came through the publication where there are the 3 different type of PTM study was done on the KLF, and this was only towards one of the KLF6. So here they have reported 3 different PTMs, ubiquitination, phosphorylation, and the acetylation. So if we look at it here, so these are the lysine, they are the acetylation, and serine are involved in the phosphorylation, while one single ubiquitination was reported in case of the KLF6. So we went ahead and we looked for the all group 2 members which involved the 6 KLF1 till 6, except the KLF3. Instead of the KLF3, there is a KLF7 that has been included. So we took the very standard approach. We identified the PTM, and then we modeled the KLF structures because the KLF structures are they are not available in the PDB. Then we took we identified the interaction partners, and then we generated the protein-protein complex along with the interaction partner. So we have the KLF and then the interaction partner, and the complex structure was generated. And then PTMs were incorporated at the selected sites that we identified in the beginning. And then we calculated the interaction energy. Here, as we can see that these are the different, these are all are the 6 KLF group 2 members, and we can see here these are the different phosphorylation as well as the acetylations that were identified. In this depiction, we can see that the KLF acetylation across all the 6 members, acetylation is depicted here with the red, and the phosphorylation is depicted at the by the blue color. Then we looked for the interaction energy. So as we can see here, this is the wild type. These are across all the 6 members. So in the wild type, compared to the PTM, in the PTM it was observed that the interaction energy was reduced. That means it was leading to the stability. So without going much into the detail, I'm just taking 2 figures from this publication. So here, in this case of the KLF4, and its interaction partner here was the EP300 complex, EP300. So when we had looked at the KLF4 EP300 complex, and there the acetylation was induced at the K52. After the incorporation of this acetylation at the K52, this complex was observed to be stabilized. The another PTM, so the first example I took was the acetylation. Here I'm taking the phosphorylation, and the same in case of the same KLF4. This particular phosphorylation at the serine 327 position, it was found to be linked to the lung squamous cell carcinoma. And in this case also, we found that phosphorylation at this particular position leads to the stabilization of the KLF4 CBP complex. So what we are observing here, it was the pattern that we observed, that whether it is the acetylation or the phosphorylation, that either of the PTM, it was stabilizing the complex. So this study was published in the Biochem Biophysics, BBA GRM basically, gene regulatory mechanism. And here we concluded that KLF PTM, they act as a molecular switch, and this switch is required based on the transition from the cancer suppressor to the promoter, because the KLF has a tendency within the same gene, they can act as a suppressor as well as the activator, depending upon the type of the cancer. It may vary, breast cancer versus the lung cancer, and on the also the stage of the cancer, which stage it is currently. Then while we were completing this story, then we looked for the interaction partners. The same time we found that KLF6 interacts with the SP1. SP1 is the specificity protein 1, and in this particular case, this study is in collaboration, and it took around 3 years of the work, which I'm not going to go over. So while continuing the post-translation modification aspect here only, I will try to highlight one simple aspect here, that in case of the SP1, which is also, which I mentioned that it is the specificity protein 1, what it does basically, it drives the expression of the genes which are involved in the polyamine biosynthesis. Now the role of the polyamine biosynthesis is important to, because it supports the cancer cell survival under the stress. And in the pancreatic cancer specifically, SP1 level and the activity, they are influenced by the stromal cell derived acetate, which is the metabolic bioproduct. Now, the acetylation mediated by the acetate ACSH2 of the SP1 at a particular position, lysine K19 basically, it increases the SP1 stability and the transcriptional activity. This study was further done into the in vivo models, but I will just show here an image showing the wild type SP1 as well as the acetylated SP1 at the K19. This is the MG simulation, and as we can see here that post acetylation there is a stability. So that means, continuing with the PTM, that they are playing a critical role, and also they are letting the complex to be stable. So I just took one aspect of it, but this story covers the several aspects, including the in vivo studies. If the students are interested, they can go through this publication at the Nature Cell Biology. The reference is given at the bottom. So the another aspect that we are trying to look is like I mentioned that KLF, they are involved in the being the suppressor as well as the activator. So they are the oncogenic also. Specifically, the KLF6 SV1, which is the splice variant, as I mentioned earlier, it is the oncogenic, while the KLF6 itself was the suppressor. Similarly, there's the other one also, KLF7, that is also the activator. So that is also found, the expression level have been found to be upregulated in several cancer, including the breast cancer, ovarian, and the lung cancer also. So we thought, okay, can we try to look for the potential inhibitors? So here we took the standard approach. I'm not going to go into the detail because the talk before me explained it much in detail. So it was the standard strategy that was followed, and we are trying to identify the potential inhibitor. In this particular case, it was for the KLF6 SV1. And here we have identified the potential binders also. Currently, the in vitro studies are going on to be followed by the in vivo studies. So before moving on to the KLF7, so this is a standard practice that we follow, and we have done it for all the KLFs that we are pursuing for the group 2. So here we check for the expression, the mRNA level expression of the KLF7 in this particular case. So we can see the control here, and this is the ovarian cancer particular cell line. We have checked it across the different cancers also. So I'm just giving one example here. In the ovarian cancer, it has been found the two cell lines that we are using currently, A2780 and the SKOV3. So this particular one is the A2780 P53 knockout. As we are aware that the P53 is the tumor suppressor. So if you remove that, that means the expression is supposed to enhance. That is the case here also, as we can see. And if we compare the RMST of the KLF7 wild type as well as the identified inhibitor, so we find that this one is the along with the ligand. So it stabilizes the the RMST here.
Excuse me, ma'am. Sorry to interrupt.
Yes,
We are
ma'am.
sort of time. So I think.
Okay, I will just try to conclude. Yeah, just give me like a few minutes.
Thank you for noting.
Okay. So as I mentioned that we are doing the in vitro as well as the in vivo studies. So I'm just giving one example of the in vitro study of the KLF7. Here we have done the dose dependent assay, MTT assay, and we can see that with the dose dependence, it is the MTT assay, it provides the viability, while the trypan blue exclusion assay is the cell death. So that means here we can see that once we increase the dose of the inhibitor, identified inhibitor, the death is increasing, and here the viability is decreasing. So besides that, we have done the other work also towards the pan-cancer expression data we are doing. Here you can see the differential expression of the KLF7, the subcellular localization of KLF7. And just to summarize the currently the work that is being going on by the another PhD student. So what she is trying to do, she is trying to look for the impact of the identified drug already. There is a mithromycin, that is a very well-known drug, on the SAT1 gene expression through the luciferase promoter reporter assay. Along with that, she is trying to understand the protein DNA interaction, how it is interacting, and along with this, we are doing the protein-protein interaction, and obviously being a structure biologist, we are in the process to purify the protein and look for the 3D structure. And with that, I would like to acknowledge that Bennett University for all the infrastructure, the funding from the DBT for 204 project, and PhD students, obviously, they are the one who are carrying out all this research. I might be presenting, but the work is carried out by the PhD scholars. Kanupriya Jha, she graduated, she completed her PhD last year, and she was the one who started the KLF story. And this is a small group, and a couple of the new students have joined, which are not in the part of the picture. With this, I conclude and will be happy to take any question.
Audience, do you have any question? No questions? So thank you so much, ma'am, for such a
Thank you.
wonderful presentation. And thank you for coming here in online mode. We hope next time.
Thank you.
now,
And sorry for not being there in person. I apologize.
We can discuss many more research with you. Thank you, ma'am. Thank you so
Yes.
much.
Thank you.
Okay. Next, also the This con this concludes our invited talks. Next, we have our oral presentation and flash talks.
The duration of oral presentation will be 12 plus 3. There will be an announcement on 12 minutes, and the duration of flash talks will be 8 plus 2. The first, let us now invite Ms. Judy Saxena and Mr. Vaibhav Singh from Parallel University, Vadodara for topic Trichogenic silver-based nanoparticles as sustainable antifungals for crop protection.
हाँ जी सर।
Please allow Dr. Hukam Joshi from the petition.
किसी ने ज्वाइन ही नहीं किया सर अभी तक। उनको बोलिए सर ज्वाइन करेंगे वो।
क्या नाम है? वो तो अगले करेंगे।
तुमने ज्वाइन नहीं किया।
कौन जॉब किया?
तुमने अभी किसको अनाउंस किया?
हाँ सर। देखिए उनके एंड पे होगा, उनके नंबर उनको व्हाट्सएप कर दे अरविंद से नोएडा वाले।
जो आ रहा है नहीं आ रहा है उसको आगे पीछे शफल कर देंगे। यू जस्ट मास्क अनुप्रिया अनुप्रिया का करा दे प्रेजेंटेशन उनका हाँ ये मैं डालू मैं तो मतलब अपना लिस्ट में सेवन ये अनाउंस कर अनुप्रिया ठीक है अनाउंस करनी
Next we have Anupriya Narukar, Monica Gualmini from Amity Institute of Behavior and Allied Sciences to present on psychological buffers against climatic driven infectious disease impacts on mental health.
This is my option. This is an adult TDS.
ये कैसे होगा ये बता दो मुझे। ठीक है
This.
कैसे कर देंगे आप?
Sounds familiar. You could use this button.
ओके
Hello, Dr. Johi. Hello, Dr. Johi, ma'am. Can I hear me?
Yes, hello, sir.
Hello, Dr. Johi, ma'am.
Yeah,
Can I
hello,
hear
sir.
me?
Hello, sir. Am I audible? Am I audible, sir?
Yes, ma'am. In between, I line up one student for presentation. So just please wait for 5 minutes, and then after we will continue with your talk.
Okay, okay, sir.
So greetings everyone, a very warm good morning to all the students, faculties and
chair over here. I am Anupriya Daruka and I am currently a PhD scholar from Amity University, Rajasthan. So the topic for my presentation is psychosocial purports against climate-driven infectious diseases impacts on mental health. So moving forward, I am basically talking about that there is a lot of climate change that is happening around the world. And because of that climate and the weather changes that has been very drastic, there has been a lot of significant infectious diseases that arose out of that climate change, which pose a lot of impact on the one's mental health, be it in a community level or on individual levels. So to cater this, a study was done. This is basically a review study where we have reviewed around, where we have basically searched and found out on a surface level 80 papers. And after that, we shortlisted 21 papers on the same line. The topic that I've chosen is basically in line with the Mental Health 2025 themes, which is access to services, which basically says that we need to cater mental health during the catastrophes and emergencies as well. Because that is the major duration where a lot of mental health changes and the crisis takes place. This has been very evident since in COVID-19, we all have been in the lockdown and we have very closely observed those mental health crises, even either we have faced or our family members have gone through. So the major psychological variables that I have taken up is resilience and social support. Resilience is basically one's ability to bounce back in a situation of crisis, whereas social support is a variable where we talk about the constant support we can gain through one another, be it in form of friends, family, peers, teachers, and as a community. So this particular iceberg is depicting how mental health disorders are the surface things that we see and that is very visible. But once we go in the deeper level, we can see that the major emphasis that comes from is the environmental degradation, the displacement that people go through as a result of climate change, and also the resource scarcity. Because whenever the climate change happens or some catastrophe takes place, be it flood or drought, a lot of population and resource scarcity takes place, which further leads to mental deterioration. So there has been various researchers have found a lot of significant differences and the role resilience and social support play in identifying mental health challenges. So it can be observed that perceived social support, resilience as an adaptive capacity can contribute a lot in dealing with the crisis than one overgoes. These are the major review studies. As I have mentioned, we have shortlisted 21 research studies. Out of those 21 research studies, these are the major 6 findings that I have incorporated in the presentation. So all of those studies have a very similar pattern, which says that once the climate change takes place, a lot of diseases arises, which further results in a lot of psychosocial impact and the psychological impacts one goes through. And when these impacts occur, for example, depression, anxiety, PTSD, these are some major disorders that takes place out of those infectious diseases. So a lot of studies have said that if we work on resilience and a social support as an aggregate capacity, we'll be able to deal with those crises that arose out of those climate change and infectious diseases. So this is the basically information about the research methodology that we have incorporated. Resilience and social support as a moderate factors that can contribute in enhancing one's mental health. Then the data source has been taken from PubMed, ResearchGate, Google Scholar, and other relevant journals. As I have already mentioned, the studies have been taken up from the year between 2015 to 2025. And out of which 21 most relevant studies from the topic have been selected. So this is the inclusion criteria. The papers were only taken out, they were in English, and even if some translated papers were available. Exclusion criteria, we have not incorporated any newspaper articles or blogs, and we have also not incorporated any researches on animals. Ethical considerations, as the paper is a review paper, no such ethical considerations were required, but due credit and citation was. Sure, sir. So these are the major findings. This is basically how climate change, infectious diseases, and psychosocial impact gets moderated by the social support and resilience, which further leads to community cohesion and adaptive capacity. These are the major themes that arose out of those studies. So yes, to conclude, the topic that I have chosen is in line with the mental health theme, and it further explains the role and of resilience and social support in moderating, and how we can build a resilient system and a social support aggregate capacity to work on these crises that ones go through. Thank you.
Thank you for a valuable presentation. Next, we have
Somebody might have question. So if anybody has a question, you can ask. Sir, any questions? Okay, so I have a question. I have shown review kind of presentation, right?
Yes, sir.
So you have selected data from PubMed, Scopus, and all these things. Have you used any software for it, or simply you just gone through this website, put your keywords and?
So basically, I have only gone through direct searches, and based on the direct searches.
So are you sure you have covered all the papers from 2015 to 2025?
Majorly, yes.
How many papers you have searched?
So it was around 100 or some papers.
Probably 2015 to now, at least 25,000 people might have published on this.
Specifically, if we are targeting resilience and social support,
So
then the number is very less.
What I'm trying to say is, there are ways of doing this kind of analysis.
Yes.
There's bibliometry analysis you can do, then there's meta-analysis which you can do.
Yes.
Prisma studies you need to follow, right?
Yes.
For this kind of work.
Yeah, that's why I only mentioned that it's basically a review paper. Okay, thank you.
Thank you. Let's now invite Ms. Julie Saxena and Mr. Vaibhav Singh from Parel University. Trichogenic silver-based nanoparticles are sustainable antifungal for crop protection.
नहीं उन्होंने मना किया इनका शक्ल भी तो कितना अच्छे से फिर ये तो ऑन है इस तरफ ऑन होती है यहाँ से बड़ा सुबह
Sir, is this visible PPT?
Yes, ma'am.
So shall I start?
Ma'am, please present it in present mode.
Is it not visible? Shall I start?
Ma'am, it is visible, but it's not full screen.
It's not. Okay, got it. So shall I start?
Yes, ma'am.
Okay, good afternoon, everyone. This is Dr. Juhi. I think it's visible. No, I think. Yeah, is it visible full screen? Okay, so myself, Dr. Juhi Saxena, from Parul University, and today I'm going to present the ongoing research work titled Trichogenic Silver-Based Nanoparticles as Sustainable Antifungals for Crop Protection, which is actually in line with the theme of the conference, where we talk about the emerging threats which can be tackled with the new alternatives for combating plant diseases. So this is the abstract that has got published and has been submitted. And I will be covering the introduction, material methods, results, and discussion in the subsequent slides. It is worthwhile to mention that nanoscience, which is actually emerged as a buzzword, has certain limitations which has to be dealt with new scientific interventions. And as we all know that the nanomaterials which are being synthesized through synthetic or chemical physical routes, they are having a lot of ecotoxicity issues. So nowadays, these nanoparticles has been explored to be synthesized by using biological routes, and through biological routes, the metal nanoparticles are very exciting. It is very important to use metal nanoparticles, which are having silver base because they are potent antifungal and antibacterial agents, as they have better interactive surface properties. The target pathogen that we have selected here is Aspergillus niger, which is reported to have black mold or the root rot diseases in plants, and this has been targeted by utilizing green chemistry using nanoparticles. And why specifically nano silver? Because this biocontrol fungi has a reported role as an antibiosis, Trichoderma Derma hertzianum, which is actually is known and is being routinely used in fields against phytopathogens. So now the idea is to replace the existing trichoderma-based fungicides with the ecotoxic nano weapons, and so as to have their better utilization. And in an attempt, we also foresee to explore the ecotoxicity with the various environmental models. So in this present study, we attempted to exploit the biocontrol fungi with the MTC3 strain 801 for the cell-free synthesis for silver-based nanoparticle synthesis. Now, with this, the present research aims to develop an eco-friendly, sustainable method. And routinely, if I would talk about trichoderma, it has a strong bioreduction and stabilization ability, as if this fungi secretes a lot of enzymes and proteins. And for example, they fall into the category of reductases, oxidases, and they have together, these proteins and enzymes together, they act as a reducing and capping agents. And the approach is very eco-friendly and cost-effective because we can even use the agricultural waste for cultivating this fungus, and the media requirement is very simple. The antagonistic role of an antifungal role of this trichoderma based proteins, enzymes, and nanoparticles is well reported in the literature. So we have attempted to optimize the synthesis of nanoparticles, and so as to have better bioefficacy. And it's a contemplation attempt to have a better nanobio synergy, exploiting biocontrol fungi for as a new nanofactory. And there is always an adaptability of this model with ease of optimizing using various statistical design. So with this presentation, we have I have tried to discuss the preliminary findings, and we have cultured this strain under ideal laboratory conditions, and initially the strain was procured from IMTech, and it has been routinely maintained and subcultured on PDA, and these plants and water oil stocks were also prepared. As per the methodology, these pure cultures being first been inoculated into the production broth, where the uniform spore suspension has been added under sterile conditions, and here we have used starch as an inducer, which is in line with our previous findings from the literature, as actually has initiated the synthesis or the increased production of amylases, which is again one of the important enzyme which has played important role in bioreduction process. So after that has been the fungal biomass has been cultured and has been filtered and then transferred to the distilled water, where through the reverse osmosis there is a secretion of biomolecules, enzymes, and proteins, and then the biomass has been filtered. Now the self-free filtrate has been challenged with the material source solution, and then process optimization has been carried out. So various techniques have been employed initially. Now we already have sent for the advanced characterization. Our samples has already been sent, but initially we have characterized our samples with UV visible spectroscopy and FTIR and TEM analysis. So the purification of particles from the sulfate filtrate is being carried out by high-speed centrifuge, and the pellet which is being utilized is further suspended in the distal sterile water for further investigations. An antifungal assay has been carried out on Aspergillus niger reference strain, which is actually the most potent phytopathogen and is an emerging phytopathogen because it has broader aspect of pathogenicity with the different plants. So this extracellular biogenic route is very impressive and is the sustainable approach is in aligned with the SDG 2, which aims for zero hunger and which also contribute towards the agricultural sustainability. And it has been well investigated previously also that these fungi are metal tolerant, and we can always have breeds of different nanoparticles which could be synthesized using this fungi. So the better idea is to have better optimization processes to have enhanced nanomaterials for various plant pathogens. And we prospected the silver-based nanoparticles from this particular strain would be new alternatives to the available fungicides, which already need further investigations with the field trials. Now, this is how we have, this is the general plate and microscopic morphology of the Trichoderma hertzianum. And this is how we have cultured it into the media and then transferred to the the white balls on this biomass is clearly visible, which we have transferred to the distilled water for having cell-free filtrate synthesis. And initial UV visible spectroscopy has been carried out. There's a transformation and change in the color, and we have a unique SPR for 420 nanometer for the silver nanoparticles, and we also have characterized for NH bands and CH bands for through the FTI that these are the biomolecules which are mainly responsible for this bioreduction process. Also, various parameters has been investigated for optimization so as to have better and enhanced efficacy. For example, the different types of media, salt concentration, different biomass, and pH and temperature. And after all, the optimized batch is then further characterized also. So we have seen that after optimization, the size has been reduced and it is scaled down to 40 to 50 nanometer. And the crystal lattice pattern has also been investigated using X-ray diffraction. So these particles are now small, good enough to interact with the fungal surface and can challenge the the pathogenic fungus and it is prospected that would have better efficacy. So now the next step, we have screened it again through the food poisoning method, the silver nanoparticles with the Aspergillus niger, and we have seen that there is a decrease in the radial growth of the fungi in the increasing order with the increasing concentration, and we have seen that this 50 ppm is an ideal concentration where we have seen maximum reduction in comparison to the positive control. So the primary investigation highlights that the silver-based nanoparticles from the Trichoderma hertzianum they are the good new novel nano weapons, and we can exploit this biocontrol fungi as new biofactory, and no external chemical capping agents are required, and the particles which are synthesized are also stable for 6 months, which we have already characterized through UV visible spectroscopy, and these remain stable and there is no agglomeration or aggregation. The particle size in the range of 40 to 50 nanometer. And initially we have screened with Aspergillus niger. Now we are also in a future aspect is that we are screening with the plate methods with the other available phytopathogens with us, and then we will be planning for the pot and field trials. We are also planning to see the effect of these nano particles in combination with other micro elements, and with this basic findings, we can utilize Trichoderma derived nanoparticles as a strong nano antifungal alternatives. And this biotemplating process is a very important step where further research is required to assess the ecological risk assessment. So these are the references for the study. Thank you.
So very nice presentation. So session is open for questions. If anybody is having queries, you can ask. Anyone is having queries?
Hi, Julie. I have a question regarding your presentation. So are you planning to apply this in the market or expand this work in future? Any future plans?
So you are asking future plans. I'm sorry, the audio is not clear to me. If you can please repeat, I'm really very sorry.
Yeah, what is the future plans of this project?
Yes. So initially we have screened the potential of this particular fungi with the reference strain, but actually here in Parul we have a collaboration with Agriculture Department. So what we are doing, we are isolating the fungal pathogens from the field, which is directly being isolated from the plants or the crops which is infected. So we are isolating the strains and then we are characterizing them, and then we challenge the same phytopathogens with the optimized breed of nanoparticles, and then we will be following the field trials. So what we are planning is to develop a nano spray. We have our collaboration here with the Agriculture Department, which through this nano sprays, what we will do, sir, we will have a control plants and we will have a infected plants, and we will spray these nano fungicides, and then it would be further, I mean, would be further validated that okay, actually this nano weapons which are actually giving good lab results could be translated to the land results.
Thank you.
Thank you so much.
So I also have a few questions. So can you, hello, am I audible? Hello. Dr. Rudy, am I audible to you?
Yes, sir, but again, sir, I'm struggling to hear properly, but I'm trying. So if you can please repeat your question. I think there's echo in the voice, and that's why I'm sorry for that. Sir, mic is mute.
एक बार बोल के देखिए.
Yes, sir. Yes,
No,
sir.
I am
Please.
audible clearly. Yeah.
Yes.
I just want to ask that why you have selected silver nanoparticle. While silver is neither macro or micronutrients for the plant, it might have a deposition impact later on in the plant, and it can also enter into food chain for some other problems.
So we have a kind of, so whatever initial investigation we have carried out, we also are planning in, though the results have not been shown here, but we have tested them in the ecotoxicity model with the algae, sir. So we have less toxicity reported here. So these are not silver monometallic, these are silver oxide. Also, sir, we are planning to develop a bimetallic nanoparticles. We are trying to club it with the copper and zinc, which are actually required as microelements with the plant. So that is, we are investigating, but right now I have not shown data here.
And how much economically viable using silver as pesticide is?
So silver cost available to the fungicide currently is, yes, you are right, sir. The cost is also attached to it. But when we reduce the amount of silver here, actually, sir, we are not investing anything in cultivation fungus. We are actually utilizing the waste from the agricultural residues, and also we are establishing this here to utilize. But yes, I cannot ignore that cost is attached. So I think with the future investigation, I would be more able to answer this question, that what would be the exact cost. Yes.
And one more question is, you have used Aspergillus niger, although you have mentioned about the Trichoderma strain, but you have not mentioned anything related to the origin of Aspergillus niger, whether it was a plant origin or anything else.
Yes. So this strain has been provided to us from the Agriculture Department here as a reference strain, sir.
It was responsible for causing certain disease into the
Certain
plant?
disease, no, not in the humans, sir. No, not in the plant. Yes.
Okay, so for stability, you could have done Zeta and TG analysis, rather you have choose to do UV SPAC. Why so?
Yes. Initial findings, we actually have sent it to the TEM only after doing DLS, zeta size and zeta potential. And for stability purpose, like we are planning that, let me first try on the plate assays, and then we are planning to go to the land trials, or once we are optimized, we are sure shot, okay, this is the breed of my nanoparticle, which is giving maximum efficacy against maximum isolates. So that particular breed of the nanoparticle would be then tried. We're planning to do thermal analysis so that we can.
One more question I'm having. This is going to be the last question. Why you have chosen submerged fermentation while fungi are usually aerobic organisms?
Yes, so for solid state also, sir, we are planning initially at the lab scale. We actually
solid state, but you could have done like without shaking. You could have done, so you will get growth on surface.
I would take care of this suggestion. Actually, we are trying to use now the agricultural waste residues also. So it is a circular economy. We will close the loop.
Okay, fine. Last feedback is please italicize the names of scientific organisms and everything.
Sorry, sir, if I missed it.
Thank you.
Thank you, Ms. Rui, for sharing your contribution towards the sustainable agriculture.
Thank you so much.
Please welcome Mr. Anupam Jyoti, Mr. Gaurav Agarwal, and Ms. Aarti Mulay from Parul University, Vadodara, presenting modulating sepsis-driven hyperactivation of neutrophil effector functions with reversatrol and in silico and ex vivo strategy.
It's fine. इनके नाम से ज्वाइन करने वाले थे मैम के नाम से बट आई शायद सेटअप कर रही हूँ एक बार बोलिए सर
एक बार फिर से कुछ माइक नहीं कर सकते यहाँ पर आवाज नहीं आ रही
Excuse me, sir, you are muted.
Hello, Anupam sir. Please unmute, sir.
Sir, I am unmute.
you're audible
Yes, sir, you are audible.
please share screen
Okay, I am audible. Okay, just give me 2 minutes. I'm just sharing screen.
वीडियो बंद कर दो वीडियो बंद कर सर की सुनिए मैं अपनी
let's enter normal
I hope screen is visible
Yes, sir.
and slides are changing as well.
Yes.
Okay, thank you very much for the confirmation. So, thank you very much, especially the organizer, Dr. Sanket Kaushik, who has given me an opportunity to present some part of my ongoing work. And it's always great to be back with the people from where I started my career. So I'm Dr. Anupam Jyoti. I've started my research journey, or you can say career, from MIT University Rajasthan Jaipur way back in 2011. And from that, I'm continuing my research work basically in the field of sepsis. So it's good to be back there and interacting with young minds. So with that, I'm starting my presentation. So title is Modulating Sepsis-Driven Hyperactivation of Neutrophil Effector Functions using resveratrol and in silico and ex vivo approach. So as we know, sepsis is a life-threatening condition from dysregulated immune response to infection, and it leads to systemic inflammation, organ dysfunction, and death if not treated properly. Almost as per the data received in 2020 from Root et al., it is almost 11 million deaths annually for the sepsis, and it is one of the third leading cause of the death worldwide, especially in the emergency department. And it has a high morbidity and mortality rate, significant impact in low and middle income countries where resources are very poor, because it is directly related to infection. So there the resources are not proper to take care of those infections. So it will lead to inflammation, and if not treated properly, it gives to the mortality. The challenges remain despite advances in the critical care, because sepsis is highly heterogeneous. The exact reason or root cause or signaling pathway or exact target protein is not defined, because patient to patient it varies a lot. So one size fits all theory doesn't suits here. So there is a lot of complex pathophysiology which lack the universal therapy. Like in case of other diseases, we have some universal therapy, but in case of sepsis, it is actually not universal therapy is suited because of the complex pathophysiology and highly heterogeneity. So if we talk about the pathophysiology, so it starts with the hyperinflammatory response followed by immunosuppression. So as when the bacteria infect or fungus or virus infect, so there is increase in the inflammatory response which go and try to combat the pathogen. And then once it is done, then it has to be suppressed. Otherwise, hyperinflammation will take place and it will cause the damage to our cells. So immunosuppression comes, but sometimes the immunosuppression is not properly regulated and the immune system gets highly depressed and it will lead to some other pathophysiology, right? So today I'll talk basically of the hyperinflammatory response. So hyperinflammatory response basically driven by hyper cytokine release, basically the pro-inflammatory cytokines like interleukin and the TNF alpha, IL-1 beta, and interferon. And neutrophils are the cells which are WEC cells which reach to the site of infection inflammation first. So they are become hyperactivated, and once they are hyperactivated, they reach to the site of infection and they kill the pathogen by internalization as well as through phagocytosis externalization. Through internalization, they engulf the pathogen and produce lots of oxidative stress with the help of certain enzymes or proteins, say INOS and RAC2. And the symptoms of the hyperinflammation sepsis basically the fever, hypotension, altered mental status, and later on organ failure. These are the current treatment options which are available for the sepsis, like antimicrobials, vasopressor as a standard treatment. So, however, in case of the antimicrobial, if the pathogen is not identified properly as well as quantified properly, so the broad spectrum antimicrobials can be given and which may lead to the development of MDR. So that will further complicate the treatment. As well as the in the current treatment option, it doesn't address the immune dysregulation, that how neutrophils are hyperactivated or immune cells are hyperactivated. So there is no as such therapy available which can actually address this immune dysregulation and target the immune dysregulation pathway. So antibiotic resistance, I have already mentioned. Organ toxicity, in some cases, it has been reported. In some of the case of the antimicrobials, there are chances of the organ toxicity. So again, it will lead to the multiple organ failure, right? Secondary infections, if a lot of antibiotics can be given and it may lead to the suppression of the immune system, low pathogen, then the chances of opportunistic pathogens are there, so which may lead to the secondary infections. And limited success has been with their corticosteroids due to immunosuppression. So that is a dexamethasone. So in the COVID case, lots of dexamethasone has been given, but yes, in some cases it has success, but in some cases it has not success, and it may lead to the immunosuppression. Again, I am saying because of the patient heterogeneity, one size doesn't fit all. So that's why we require a targeted pathway specific targeted protein which can be targeted. So yeah, we talk about the neutrophils. So neutrophil are the first line of host defense. As I mentioned earlier, it internalize the pathogen. So these are the, you can see this is the nucleus multilobulated, and this is the granule protein. So myeloperoxidase, MPO, and then elastase are there, which is a xerophilic granule. This is a specific granule, and this is the enzyme NADPH oxidase, which produce lots of the reactive oxygen species inside the cell as well as outside the cell also, right? And it sometimes releases the granule protein, which is called as degranulation. So it produces the reactive oxygen species as well as nitric oxide also produced with the help of inducible nitric oxide synthase. Apart from that, both nitric oxide as well as the reactive oxygen species, they are produced in the close vicinity. So there are the two protein, that is INOS as well as RAC2, which is a part of NADPH oxidase. During the case of infection inflammation, they come close to each other and they interact, which we have shown in our previous work, that they physically interact and their product also gets interact, like INOS produced NO and RAC2 produced O2 dot minus. They interact and produce a peroxynitrite, which is a very highly potent radical, right? And this radical kills the pathogen, but if it is not properly regulated or quenched, it may lead to the killing of our own body cells. That is a case of cytotoxicity. And the other mechanism, that is neutrophil extracellular trap, in which neutrophil, in the case of pathogen infection, neutrophil expels its chromatin. So here you can see this is the chromatin which is coming out from the neutrophil, and they are going to trap or the pathogen, say here pathogen in green color, bacteria I am showing here, and these are the granule proteins. So they also come and kill the pathogen. The histone are one of the key protein. So we are knowing that histone is basically structural protein of the chromatin, but in the last 5-10 years it has been reported that histone is a very good antimicrobial in nature also. So histone comes outside along with the DNA and it goes and kill the pathogen, right? Okay, in our work we have chosen the resveratrol. So resveratrol we have actually is a natural polyphenol in the present in the red grapes. It has a protective effect in the various diseases, basically the cancer, CVD, diabetes, neurodegeneration, aging, and many more. And because of its antibacterial, antifungal, antiviral, it shows a very good potential in curing the sepsis, basically for quenching the free radical, doing the anti-inflammatory activity. So it has a good potential, right? So with this background, the present study we have designed is first we will go with the identification of the hub genes involved in the sepsis related pathway using in silico analysis of GEO dataset. So as I mentioned earlier that it is basically the highly heterogeneous. So pathway specific target should be identified. So that we have designed with the first objective, then some molecular docking experiment for binding resveratrol with identified hub gene, then evaluate the potential of the resveratrol to modulate the critical pathways implicated in the sepsis. And these are the methodologies which we have employed. Like, so I'll just show you in the form of the graphical abstract. So first we have taken the GEO data set and then identified the differential express gene using the R. Then we have identified the common gene identification with some in silico tool, then gene selection, annotation, and functional role. Then we have created the protein-protein interaction network using the string database, then hub gene identification with the cytoscape, then functional and enrichment analysis. Then we have done some molecular docking experiment, and to validate this, we have done some ex vivo experiment in the human sample. So I'll discuss all details later on. Okay, so initially we have chosen the 3 data sets which are available in the GEO, and these data sets are basically sepsis versus healthy volunteers, right? So approximately is the data set of 250 or 350, nearly 350, and it is a data set of whole blood, and some are microarray and RNA-seq data set. Then we have processed the data set with the help of inbuilt GEO2R, which is a GEO-R language already processed there. And we have used the volcano plot and the mean difference plot to differentiate or to show the data. So we have here used the functional enrichment as well as the fold change more than 1.5 to identify the differential express gene, mainly the genes which are going to be upregulated we have chosen. And then we have done some Venn analysis. So from all the 3 data sets, what are the DEGs and what are the common DEGs? So almost we found nearly 1313 DEGs are there. And then we have done some scoring and we found that almost 9 DEGs are there which have been showing the score more than 0.4 as by default setting. So we have chosen those 9 genes, and then we have done some network analysis and hub gene identification and showing with the help of the MNC algorithm and CytoHubba plugin. So we found that gene which is S100A12 is a critical gene or which is in the central part. And then we went to the literature and we find that, okay, this S100A12 is a gene or its protein part is highly its role in the inflammation in basically in the WBC cells. So it is actually matching with the whatever the pathophysiology of the sepsis is there. There also the white blood cells, especially neutrophils, are there, and this S10012 plays important role in the degranulation as well as the chemotaxis of the neutrophils. So we thought that with this simple in silico analysis, we got the right target protein, right? Then we have done some cake pathway analysis, and then again we found that, okay, yeah, as per literature, it is showing it has role in the neutrophil degranulation, as well as it has some role in the TLR 1, 2, and 4. So TLR 2 is basically for the gram positive and 4 is the gram negative. So we thought that, okay, so this protein is basically playing inflammation role in response to both gram positive and gram negative. So for further validation experiment, we have used both E. coli as well as S. aureus as a model pathogen to mimic the sepsis condition for gram positive and gram negative. Then we have put this SA1002 into the HERB database, and we found that there is a drug which is called a resveratrol. It is showing the maximum potential to be inhibitor of this. Then we have done some molecular docking experiment, and we found that okay, resveratrol and S100A12 has a binding roughly -7 kilocalorie per mole, and so it is showing okay, it is has a moderate activity, and the molecular dynamics and simulation experiment is ongoing actually to further validate whether the binding is in the stable binding or not at 500 nanosecond. So that is an ongoing process. So that's why I'm not able to show the data here. Then with the wet lab experiment, because all the in silico work is basically for the prediction and all, right? So it needs to the validation. So first we have taken the approval from the Parul University, and then we for the handling of the patient sample. Sorry.
Sorry,
Hello.
but we are running, sir, sorry to interrupt, but we are running late, so sir, can you please conclude?
Sorry, I can't hear you.
Sir, we are running late on time, so can you please
Okay,
conclude?
okay. So I'll try to finish. Okay.
Thank you, sir.
Okay, thank you. So first we have to optimize the concentration of resveratrol. First we have done the RBC toxicity assay, which is a very standard assay, also called as the RBC hemolysis. So in the left side A, B, C, D figure, you can see in the different concentrations of the resveratrol. As per the literature, we have chosen from 10 micromolar to 100 micromolar. So up to 50 micromolar in the right side in the blue color bar, you can see there is no hemolysis, and on the right side in F figure, you can see the little redness in the tube, right? And at 100 micromolar, so almost all the cells have been lysed. So since it is started from the 75 micromolar, so we have chosen just below the 75 micromolar, 50 micromolar is a safe concentration. And we try to evaluate whether at this particular concentration, resveratrol is showing antioxidant as well as anti-inflammatory activity or not. Then we isolated the neutrophil cells and incubated with the resveratrol at different concentration, and we have challenged the neutrophil with the E. coli. So here, oh sorry, concentration was 50 micromolar, which we have optimized in the previous toxicity assay. So you can see here in the A graph, this is the control neutrophil cells and the cells treated with the resveratrol. So almost no effect of resveratrol in terms of toxicity. And then we have challenge with the E. coli. So level of the free radical, specifically the reactive oxygen species, it increases in the presence of E. coli just to mimic the gram-negative pathogen infection in the cells. And then in the presence of resveratrol, it has reduced. So it is showing some, the antioxidant potential or quenching the free radical, which is a critical for the pathophysiology of sepsis. Same type of data we found in the S. aureus, which is again a gram-positive bacteria, right? And we have taken here as a positive control is a PMA. PMA is a forward myristate estate. So this is a protein kinase C inhibitor, which induces the ROS with the help of NADPH oxidase. So similar type of data we have found in the case of the PMA as well. And then we went for the nitric oxide generation. If you remember my previous slides where INOS and reactive interaction I have shown. So NO and O2 dot minus, then peroxynitrite. So in previous slide, I have measured here the superoxide, and this slide I have measured the nitric oxide. So more or less similar type of data we have observed in the case of E. coli as well as in the case of S. aureus. Then we went for the very classical experiment, that is for the anti-inflammatory assay for the neutrophil extracellular trap. So in previously I have shown you about the nets where the neutrophil throws its arsenal outside the cell and then try to capture the pathogen.
Sorry to interrupt, sir, but we are running late on time.
Sir, I am just concluding. This is the last slide. So this is the neutrophil, and this is in the presence of PMA. You can see some thread-like structures are there, which is coming out from the cell. These cells have been labeled with the cytoxine. This is the positive control DPI I have used, which is a known inhibitor of the PMA. And here is our test compound, resveratrol at 500 micromolar, sorry, 50 micromolar. So here you can't see any thread-like structure, almost similar to that of the DPI, or you can say the control, right? So here multilobulated cells, you can see that is the showing about the conformation of the neutrophils, right? So with that, I am concluding resveratrol exerts through the multi-targeted mechanism, reduces neutrophil oxidative burst, inhibits the chromatin decondensation and nets formation, modulate oxidative and nitrosative signaling, attenuate the pathology of the sepsis, decreases the RBC lysis resisting vascular protective effect also, indicate potential role in endothelial and erythrocyte interaction. Okay, so that's all from my side. These are the references which I have used. I'd like to acknowledge the funding body, DST, SERB, and ICMR, and GSBTM. GSBTM is the DST of the Gujarat, which I have got the project also, and obviously the healthy subject from where I have taken the blood sample. So thank you very much. Sorry I have taken much time, but I will be happy to interact or any question. Thank you.
Now session is open for discussion. So if any query, quickly ask questions. Thank you, sir, for wonderful session. And I wish we could share some more time, but maybe next time. Thank you so much, sir.
Yeah. Thank you, Ravi Ranjan, sir. Thank you. Thank you very much. And thank you all for patient hearing. Thank you.
Thank you, sir, for a wonderful presentation. Next, we have Mr. Pramod Kumar Gupta from DY Patil University, Navi Mumbai, on topic screening of compounds for wild and mutant EGFR using integrated machine learning and molecular modeling techniques.
Just. No, this is not mine. This is mine. Okay. You okay? Yes.
Good afternoon, all. Am I audible? So my topic is screening of compounds for wild and mutant EGFR, and we are using integrated approach of machine learning and molecular modeling techniques. Basically, it's a completely in silico work. The content. Oh, yes. Display. First, swap using to use need. Display the second.
नहीं हो रहा क्लिक ही
एक मिनट This one?
So the options are not available. The options available there.
पहले किसी का डिस्प्ले हो रहा है क्या उसको चेक करो कोई और प्रेजेंट कर Yes. Thank you. So content we are going to discuss, my team, is the identification of EGFR based known and investigational compounds which are already available in the database by using a derivative mining techniques. We are using cluster based technique over here. Selective ligands predict and target prediction, protein-protein interaction using network analysis. And the third part is molecular modeling and simulation-based study with this selective compounds. And finally, we are also working on the allosteric docking. So these four things we are going to discuss now. So most of us are working with EGFR because of very exclusively work protein in terms of inflammation, cancer, infection. It has a plenty of role, but there are plenty of mutation also that has been identified in the series of years in the last few decades, which is causing a serious drug resistance. And this mutation, because of the co-evolution process, some mutations are within the binding site and out of the binding site, and that is stretching the binding site region, not only the ATP binding site, but also the allosteric, which is quite close to the genuine ATP binding site. And we are going to observe this mechanism over here. There are plenty of mutations, plenty of overexpression types studies already known. So mutations in various exonic regions in 9, 10, 11, 12, 15. So there are plenty of mutations and few mutations that we are targeting here. Obviously, here I'm just presenting few mutations. Still, my lab is working throughout plenty of mutations we are working over there. So this is the site that I'm discussing, that ATP binding site and the allosteric, and you can observe they are quite close to each other. And that's what we are looking in the initial layer, working with the ATP binding site interaction.
And then we are also working with the allosteric. That means at a time, two molecules, compounds, or you can say experimental or drug, known drug, will be interacting for the better efficacy that we are predicting. So selecting a scaffold, that is, there's plenty of molecules in market, but still we are not able to conclude a very good one. So my lab is already working with benzimidazole for a very long time period. So we work on benzimidazole. Why benzimidazole? Because it has a diverse activity. It has a very diverse activity in diabetic. It goes with the PPR gamma. It goes with the EGFR. Even we worked on the c-KIT kinase and CDK2 we have worked. So we have selected this, and obviously the data is present with the benzimidazole. So identification of this data, because there is a very small space has been there right now to extend the chemical information, because chemical space is restricted because molecular weight goes beyond 500. So it loses the activity of lead compound like Lipinski filter, Verbose filter. So we have to take care about it. So we are looking for some small molecules which are already there in the database. So we can now it's only started right now. I was yes. So we have already using the machine learning technique. So we have generated a library by using Clevin's with the clustering technique, and the input was all the physicochemical properties with the log p value, Lipinski filter, structural features. That means a single ring, double ring. All these features were considered, and we have screened some compounds. And from that, we have compared with the known inhibitors which are there into the database, like FDA approved drugs. That's what we have matched over here. And this is the entire flow of our techniques for the machine learning that we have given. We have selected the benzimidazole scaffold, and from the data we have considered IC50 value between 1 to 10 micromolar, and then we have retried the drugs, all the chemical compounds, which is around 4,000 plus compounds. From that we have compared the FDA approved, and we got some EGFR inhibitors. We have compared, and finally we land up with these set of compounds, which is closely associated structurally and physiochemically. Apart from other derivatives that we have identified, this is quite close to benzimidazole. You can see this ring. So not exact, but yes, very close structurally. So this is one, but like this, we have got 5 to 6 compounds with all the FDA approved drugs. So we have screened up to 18 FDA approved drugs. In case are derivatives, these are all the derivatives that we have come with the very specific, less toxic and experimental. So this is what my completely concludes over here. So we have got more than 20 compounds in hand, and we go for a systems biology approach, that is identification for each compound. We have identified a target. More than 100 targets were identified, and after that we have just went into the protein-protein interaction network. Why protein-protein interaction network? So that we should identify the late failure, because at the end, if we find that my molecule is little bit toxic somewhere, that comes to 0. So here this helps us to identify where my molecule is working, which target, which network, which metabolic pathway, whether I should approach that or not. From that, we went to the pathway enrichment analysis, and we found certain protein molecules which are very specific targets. So EGFR has the maximum ranking over there. We are already working EGFR, c-KIT is there, SRC kinase is there. So even non-kinases were there, few like COX is there, because it comes into inflammation. Definitely we get a COX-1, 2 over there. So we perform molecular docking, and we gone with the MD simulation. Now I will just conclude in 2 minutes. This is all the results. So this is all the results. It's a thousands of information that we got. And this is star ball of protein that we say specifically with the protein interaction network. There's some enrichment with the pathways that we identified. And this is derivative 2608, that is what we have landed up with a very good interaction. Finally, I'll show you the best part. Just one more slide, because this is always your network, what I've just spoke, and there's a crunch of time. The best part we didn't gone with the partial structure, we modeled the entire length of the protein. And then we perform the mutation, and we are looking the simulation of the entire set of protein. Most of the times we make some mistake with the only going with the kinase domain. So sorry, we are not doing that part. We are going with the full length of protein, the whole length. So we have a solid information in future also. This is all simulation. There's ample of information, but there's all information. This yes. So our overall conclusion is this part, that with respect to individual ATP binding site also, and with respect to allosteric point. So we identified few molecules which are giving very good result at the docking level. And recently we have completed up to 200 nanosecond simulation also. So with the single, we have come up to 500 nanosecond. And with the double, it's in process. Yes. So I'll show you that final result is over here in the 2D view. Yes. So this is our one molecule, and this is another molecule with the ATP binding site. This is our allosteric. This results we have. So at a time, we can interact two molecules at both the sites, and it can give us a better efficacy. Because most of the time, we have seen that if you have overexpression also in the wild type, they fail in the inhibition of the cancer. That's the overall we are doing. We are working with wild also and mutant also. Thank you. That's all from my side. Yes, it's a pen drive. Questions are welcome if any. They can connect with you during the yes, people we are already looking for. Yes.
Thank you so much, sir, for your wonderful presentation. Next up, we have Hriday Ramesh, Pothumari Gupta, Malaprabha, Devajani Das Gupta from DY Patil University on the topic screening and identification of proteins involved in inflammation and breast cancer using integrated bioinformatics and machine learning approaches.
I request all presenting members to please conclude on time, because we are running short of time.
This is the one.
Good afternoon, everyone. My name is Hriday Ramesh, and I am presenting on screening and identification of protein involved in inflammation and breast cancer using integrated bioinformatics and machine learning approaches. So, how can inflammation cause breast cancer? On the left panel, you can see that normal breast tissue, suppose there is a chronic inflammation that is initiated in a normal breast tissue from several types. Hypertrophied adipocytes and crown, if you can see the crown like structures are the most prominent focus that secrete multiple pro-inflammatory cytokines, including chemokines and adipokines, which promote chronic inflammation. Other than that, the microbiome, dying or injured cells, lymphocytes, and fibroblasts, which are shaded, indicate that they are present, but they activate only when there is a promoting chronic inflammation, due to which it can cause a breast cancer. The left panel, as you can see, the breast cancer, it's a breast cancer tissue. So multiple cellular processes are present in breast cancer promoting chronic inflammation. The infiltrating lymphocytes and macrophages, cancer-associated adipocytes, and crown-like structures, mesenchymal stem cells, cancer-associated fibroblasts. These all secrete multiple pro-inflammatory cytokines such as chemokines, adipokines, etc. And these cells interact with the immune cells through MAMs, that is your micro associated molecular pathways, and your DAMs, that is your damage associated molecular patterns. So the aim of this particular project was that to identify the key proteins that serve as molecular links between chronic inflammation and the breast carcinogenesis. The objectives over here, we have used UniProt KB, that is a UniProt knowledge base, for the data extraction. So from there, we took inflammation, we used the keywords as inflammation, inflammatory related terms, and we segregated the two data set as non-protein kinase and protein kinase. The second objective was to use these two clusters as a protein-protein interaction network using string database. So here we took the confidence level as 0.9, and also we used k-means clustering as an machine learning, unsupervised machine learning technique. The third was the functional annotation of these selective proteins and were compared with publicly available transcriptomic dataset to validate their functions. So these are my materials and methods. So the protein data was downloaded from UniProt KB. They were segregated as 610 protein kinases were available and 267 non-protein kinases were used. These 610 and 267 protein and non-protein kinase were further used in string database for protein-protein interaction. So out of that, when we did the 0.9 confidence level and k-means clustering, so we got protein kinase from 610 to 149. And same way, the non-protein kinase, which were 267, we got it to 83 non-protein kinases. These further, with the help of David database, we use their functional annotation to check which all non-protein kinase and protein kinase are involved in both inflammation and breast cancer or any related cancers. So these are the string database data of the whole 414 protein kinase. Here a special mention. So while we had 610 protein kinase, while using the only confidence level, we had 414 protein kinase. This is without k-means clustering. Same with, we had 178 nodes with 0.9 confidence level, again without k-means clustering. After using k-means clustering, we got the 149 nodes and 83 nodes for protein kinase and non-protein kinase respectively. So these were some of the 149 genes, or you can say the 149 nodes of protein kinase, and the KEGG pathway, like what these genes are, in which all pathways they were helping out. So as you can see, so there are pathways in cancer, breast cancer, also in chemokines, or you can say in neutrophin signaling pathways. Same way for non-protein kinase also we had taken David database. So To conclude, from protein kinase, these three genes were seen that had all the they had the role in interleukin pathways, other cancer and also breast cancer, that is your GSK3B, MAPK1 and MAPK3. And the in non-protein kinase, NFKB, IKBK.
So we are running short of time. So sorry to interrupt you.
Yeah.
We are moving to next participants. Please allow us.
Yeah.
Thank you so much. You presented a nice presentation. Thank you so much.
Next, we have Ms. Sanchita Singh from CSIR, National Botanical Research Institute, Lucknow. She will be presenting on eco nano insecticides from aromatic plants, experimental and computational evaluation against Phenacoccus celopsis.
Hello everyone, am I audible?
Yes, ma'am. Yes, sir.
Okay, perfect. So I'm sharing my screen. Please let me know if it is visible.
Yes, ma'am. The screen is visible.
Assistant Professor, Department of Botany, Sanskaram University, and this work was done at CSIR India, right, Lucknow. So I'm starting.
I'm a presentation.
Yes, so I'm starting with the presentation. The aim of the research is to develop a sustainable management strategy against cotton mealybugs. So cotton mealybug is one of the major pests of cotton. So all the 9 states of a country, which are cotton producing states, have very high infestation of cotton mealybug. And the problem with this particular insect is, first, as you can see in the picture, the waxy coating which is present on the which is present as the exoskeleton of the insect. And the second problem is the high reproduction rate. Like 10 to 12 generations, it produces 10 to 12 generations per year, which make 5 eggs, each of them. So the current method, the problem which is caused by this particular insect is it makes your plant, the cotton plant, nutrient deprived. Then the plant dries, there's a late ball opening and the retarded growth. And as we hear is the by the farmers. So 40% of global crop yield is lost every day, every year, and over 600 millions of US dollars are spent on crop protection. And a value of insect-related damage is expected to be $200 million US dollars. So there's an urgent need of developing environment-friendly approaches. And as you can see, the cotton mealybug, this particular insect, has always managed to stay in the headlines. Now, moving towards the environment-friendly quick fix we are proposing in this research. So what we have done here is, if I give you an overview, what we have done in this particular work is we have taken biopesticides, which means we have, and then we have compared their insect mortality efficacy with the currently available chemical pesticides, and then we have done the insecticide efficacy evaluation. So moving forward with the details of all the chemical pesticides used in this particular study. So this table shows you a list of all the 7 pesticides, chemical pesticides, and their mode of action and the coding which is used in the research. Then this particular table, the next table showing you the details of all the plants which are used in this study, and the chemical and the essential oil extracts were isolated from these plants, and here are the details of these plants. Check the composition of essential oil, and this was performed by gas chromatography mass spectrophotometry. So the GCMS was performed for this particular experiment. Yes. So this is the heat map which was generated with the results of GCMS. So essential oil and were analyzed and GCMS of for the phytochemical profiling. So the major component, as you can see, was the linalool, gerinol, gyrinol, acetate, and the estrogol and pipертone. So the 6 plants which were selected, two of those plants were mentha and the Osminum basilicum, the tulsi plant. So the highest amount of estragol, which is a very well-reported insecticide, was found in one of the varieties of tulsi, which was Osminum basilicum SA, which stands for Savita. And then pipертone, which is also pipертone oxide, which is also one of the very important or very famously reported insecticide, was also found in good quantities in Mentha arvensis. So the cluster analysis shows that the oils were grouped together by dominant compounds, and there was a clear chemotypic variation. So this shows that, yes.
Sorry to interrupt, ma'am, but we are short on time. I would like to request you to conclude your presentation.
Yes. All right. So these were the bioassays which were performed, and it was seen that the insecticide, the green pesticide was giving results which were comparable to the chemical pesticides. So further, we did few of the in silico pharmaphytoceutical analysis and the TEM. So this slide is showing you the results of the TEM, and this showed that all the nano emulsions which were made were well dispersed. And this was the data of in silico phytopharmaceutical analysis, which further showed the ADMET was performed, and it showed that this was low level toxicity. These compounds were showing low level toxicity. Then Protox was performed. These were the 8 major components.
Thank you, ma'am. Next we invite Ms. Satakshi Kumari, Ms. Anjali Goswami, and Mr. Vivek Pratap Singh from NIMS University, Jaipur, to deliver their talk on harnessing herbal synergy, phytochemical and therapeutic study of the Datura metal and Desmora purpura. She is not presented. We can continue to next presentation. Next, we welcome Ms. Vishakha R. Chakole and Dr. Priyanka Sen from VIT Vellore for their work, expression and verification of HIL6 and its characterization.
Is she there?
They will be joining us online.
Vishakha. In the meantime, announce the name.
Vishakha, can you hear us? Vishakha, can you hear us?
Hello, sir. I'm here.
Yeah, please start with the presentation.
Now, Hello.
Vishaka, before you proceed, I would like to tell you we are running short of time, so can you please conclude it in 5 minutes?
Sure.
Okay.
Are the slides visible?
Yes.
Okay, fine. Good morning to everyone present here. I am Vishaka Ravindra Chakole, a research scholar from VIT Vellore, and my guide is Dr. Priyankar Sen. And today I will be talking on the topic expression purification of human interleukin 6 and its characterization. So moving further, first I will be talking about the problem statement, then objective of the objective, and then introduction, methodology, and the result. So the question is, can we express the recombinant proteins without IPTG induction? Then the answer is yes, we can produce the recombinant proteins without the IPTG induction. So what is the reason that we will be replacing IPTG instead of NaCl? So, and second problem faced by industry is like they are using tag proteins, and in our case we are using tagless proteins and with NaCl induction, not with the IPTG induction. Because NaCl, because IPTG is having so many side effects, so we will be using NaCl for the expression. So these are the objectives of this work. So moving further, so why I have chosen human interleukin 6? Because human interleukin 6 plays a vital role while immune system regulation, as you can see in this graph. As you can see in this diagram. Moreover, means it's help in many various therapeutic drugs, COVID-19 and so on. But the problem is when it is overproduced in the body, it can cause many autoimmune diseases like rheumatoid arthritis, as well as it can also be the leading cause of cancer and all. So the market price for human interleukin is now so much high, and so we are trying to produce it in the recombinant by recombinant DNA technology with so that we can reduce the cost of IL-6. This is the aim of this study. So the method used for this is like this is pcDNA 3.1. This is the mammalian expression vector. We have done the we have synthesized the primers, and with the help of PCR we have amplified the IL-6 gene. Then we have incorporated it in the bacterial plasmid, that is PRZ-B, and with the help of restriction digestion and then ligation, then transformation was done. After that, induction was done with the help of NaCl, and then expression of protein was done successfully, and then we carried out the purification for that. So this is the result showing the amplification of IL-6 gene followed by cloning. Then we have done the transformation. The transformation was confirmed with the help of colony PCR. This was the result showing the expression of protein with the help of salt-inducible system. This is the IL-6 gene, and this is ladder. This is host. This is host plus plasmid without the IL-6 gene, and this is the IL-6 gene with induction one, with different Rs. So, and this is the purification which we carried out. This was the affinity purification which was done. And then for further purifying it, we have done the side distribution chromatography where we were able to purify the protein. And then Western blotting was confirmed to confirm the IL-6, presence of IL-6. We further also did the MALDI TOF and MALDI MS-MS. This was this, we confirmed the molecular weight of IL-6, that is 30 kilodalton in our case. And this is the sequence by through MS-MS. We confirmed the sequence of IL-6, and almost 56% sequence coverage was confirmed. So to conclude this talk, I would like to say that in this way we can produce the IL-6 in this salt-inducible system, and which can be economically very favorable for the environment and for the industries too. Moreover, we are producing salt-inducible system, and that is tagless. So that is more beneficial for the industries to save the money also and to save the time also. And but if we are producing the IL-6 in this way, then we can produce the IL-6 kit which can be useful for the treatment of various patients, and economically also it will be favorable. So this is our team, me and my guide and my team. Thank you so much everyone, and thank you so much to MIT University for giving me chance to present my work. Thank you so much. If any questions you can please ask. Thank you.
Thank you, Vishaka.
Thank you so much.
Next up, we have Kiran Chaudhary, a close alum from Vanastali Vidyapith for climate and contaminants as driver of plant stress and pathogen emergence, a hidden link to infectious disease ecology. They will be joining us on.
Kiran Chaudhary, this one. Put Kiran Chaudhary का नाम क्या है? Allow. रवि सर, ये किरण चौधरी का वहां से करना पड़ेगा।
Hi Kiran, can you hear us? You may start with your presentation.
In the meantime, you can call the name of family hospital. Sir, next. हाँ, ये देखो।
Kiran Chaudhary, can you hear us?
Yes, sir.
Please start with the presentation.
Yes, sir. I'm trying. Actually, my screen is not sharing.
Okay. And
Sir, my slide is visible.
yes.
Good afternoon, everyone. Myself, Kiran Chaudhary. I am a research scholar at Vanasli Vidyapeeth and at Department of Bioscience and Biotechnology. So my topic is how climate and contaminants together affect the pathogen emergence and responsible for pathogen emergence. So my topic is climate and contaminant. Yes, sir.
Can you please show your presentation mode on the slide?
Sorry, sir.
Kiran, could you please switch to the slideshow mode, the presentation mode?
Ma'am, 1 minute.
Kiran, you have to go. Please continue.
Kiran, you can continue like this only, it's okay.
Okay, ma'am. So what are the importance? We all know that plants are very important in our ecosystem because they provide oxygen and food, and they also important for the survival of pollinators, and the plant are a major source of some medicines and necessary fibers. So with all information, we can say that plants are very much important for our ecosystem or for our healthy ecosystem. So what are the global threat to plant health? First is the climate change. For example, climate change like rising temperature, elevated CO2 level, and altered rainfall, they all are affecting the pathogen, or we can say influencing the pathogen. And some chemical contaminants, it may be like some heavy metal or some pollutant or some pesticides, they are also affecting or responsible for the emergence of new pathogen. So in this presentation, we talk about how the combined stress, for example, some contaminants and climate stress, both combinedly affect the or responsible for the emergence of new pathogen. So first is the climate change and urgent environmental crisis. So how climate change affecting our environmental. So the high temperature, which is also affect the photosynthesis activity, and some drought and in flooding condition. They also are the significant challenge for our healthy plant growth. So higher the CO2 concentration, they also affect the nutritional content of agricultural crop, and accelerating temperature also affect the responsible for pathogen mutation. So with the help of these mutation, pathogen become available, or we can say become more favorable for the new environmental condition. How the pathogen evolve their behavior with time. So higher the temperature, so what happen in high temperature condition, pathogen start to secrete some extracellular polymeric substance. So with the help of these some bacteria start to secrete extra polymeric substance. So with the help of these EPS, they increase their virulence activity, or we can say cause disease causing activity.
ये तो दूसरा है ये दूसरी है हाँ
So in condition
Online
of
इसको announce
warm
करो,
and
उसको बुला
humid
लो।
condition,
ये नहीं आई
some
है।
fungus also show their higher activity, or we can say the higher the sporulation activity. So this phenomenon of multiple infection cycle become possible within a single season. So shifting pathogen distribution, how the pathogen shifting from one area to the other area. So pathogen are increasingly migrating into new area, or we can say uninhabited regions. So some example are like Phytophthora infestans, which is causing late blight of potato, and Xyleria fastidiosa. These are the disease which is shifted to the new area. These are the example disease which shift to new area. So as pathogen shift to new area, the new host pathogen interaction start. The impact of extreme weather events. What are the impact of extreme weather event? So like flood, these flood create ideal condition for soil pathogen proliferation, and drought condition, they weaken the immunity of plant and animal, or we can say plant and animal decrease their immunity in drought condition or in unfavorable condition. So some unpredictable outbreak arise due to these irregular weather pattern. Contaminant and plant stress. How contaminant and plant stress affecting plant growth. So heavy metals like cadmium, mercury, arsenic, they all disrupt the plant metabolism, and some metal and pesticides also hinder or affecting the root growth and pigment production. So these induced oxidative stress is responsible for ROS generation, or we can say reactive oxygen generation in plants. So heavy metal, these are the silent killers of plants, So sometimes lead replaces the calcium, and we all know calcium is responsible for the membrane structure or membrane activity. So when lead replaces the calcium, membrane structure or cell membrane start to get damaged. And cadmium also poses a serious threat for plant, because it inhibits the synthesis of chlorophyll synthesis in plant, and mercury also disrupt electron transport chain. So these are the result of these heavy metals, or we can say contaminant, that affect the plant growth, and they causes some symptoms like chlorosis, stunted growth, and
Sorry for the inconvenience, but we are short on time, so I would request you to conclude your presentation.
yes sir.
I request you to conclude your presentation as we are short on time.
Okay, sir. So the what are the danger of pesticide exposure? So these pesticides kill the beneficial microbes from the plant, and some they also affect their nitrogen fixation activity, and they weaken the immunity of plant and antioxidant defense system under stress. So under stress condition, antioxidant defense system activated in plant. Some enzymes like SOD catalase, superoxide dismutase, POD peroxidase, these all are activated in under stress condition. And impact of combined pollution and heat stress also affect the enzyme activity.
So the power of synergistic effect. So synergistic effect is a condition when two type of stress together affect the plant. For example, when the climate factor like heat and drought and contaminant heavy metal, they together affect the plant, so their effect is more greater than either one stress. So these are called synergistic effect. For example, lead and high temperature show higher fungal infection. So when yes sir.
Can you please conclude now?
Yes, sir. So these are some anthropogenic drivers: global trade and deforestation and industrial pollution, and so some impacts on agriculture of these stress, so-called crop yield decrease and quality and quantity decrease, and some significant financial challenge also faces some.
We are concluding with the presentation. Yes, 5 minutes over. We are concluding with the presentation.
So with this, I conclude. So climate change and pollution are quietly putting plant under stress. So they combinedly putting plant under stress. So these stress make plant weaker and give more chance for disease to spread. So healthy plant are essential for food, clean air, and balanced ecosystem. So we need to protect our environment, reduce contaminant, and support sustainable farming.
Strictly 5 minutes. Otherwise.
Okay, ma'am. Thank you, ma'am.
Next up, we have Anjali B. Thakur from Vidyapeeth from Sardar Patel University, Gujarat. The topic is plant-derived nanomedicine, green synthesized silver nanoparticles with broad spectrum therapeutic action. She will be joining us on the
Anjali, are you there?
Yes, ma'am, I am here. But
Kindly
I am not sharing my slide.
Okay, you are not able to share your slide.
Yes, ma'am. Already someone is sharing.
Okay.
Okay, thank you.
Kiran, you have to close. Okay. Anjali, you have only 5 minutes, so kindly conclude it in 5 minutes only.
Okay, thank you, ma'am. Thank you, ma'am. Okay. Good afternoon, all. Today I am represent my topic on the plant-derived nanomedicine, the green synthesized silver nanoparticle with broad spectrum therapeutic action. Nowadays, cancers cases increase day by day. That's why we need to alternative some medication for the treatment of the cancer. So the plants is the alternative medicine for the treatment of the cancer. Plant is one of the major group of the living, vital role for sustaining the biosphere around the origin. It contains primary and secondary metabolites which are with specific activity, which are used for anti-cancer, antimicrobial, and all the activities, and in which we also we also manufacture some nanoparticles from using plants secondary metabolites which are used for the medication, electronics, and environmental remedies. I choose two different plants for my work, Polythenia and the Maduka. Then my objective is synthesize plants and then characterization by different spectroscopic method, then evaluate its activity, antioxidant and anti-inflammatory activity by different assay, and then I check the anti-cancer activity and antimicrobial activity. First of all, these are my methodology: collection, then phytochemical, then antioxidant activity, then synthesis and characterization of nanoparticle, and then antioxidant, then anti-inflammatory and anti-cancer activity. These are my phytochemical data, which indicate both plant contain different phytochemicals with different concentration. Then I quantify such amount. Then I make silver nanoparticle with both extracts, and then characterization was done by TEM analysis and spectra analysis, then XRD analysis. Then I also check particle size and zeta potential analysis, and I got result after the processing of both extract. I silver nanoparticle with good particle size and good zeta potential measure. Then I check the antioxidant activity by DBPH and H2O2 assay. Then I getting a good result compared to standard. Then I also check the anti-inflammatory activity of both nanoparticle. I got both good results. Then I check antimicrobial activity by Agarwal diffusion method against the different gram-positive and gram-negative bacteria. Then I check anti-inflammatory activity against the lung adenocarcinoma cells. Then I also get good result after the treatment of IC50 value of the nanoparticle to A549 cell. I getting good results. Then I check ROS generation. After the treatment of the nanoparticle to the lung carcinoma cells, it induce the ROS generation and induce apoptosis in lung cells. My work concluded the plant-derived silver nanoparticle are making excited stream in field of the biomedicine. Their eco-friendly production and wide range of the bioactivity, particularly noteworthy. With strong antioxidant, anti-inflammatory, antimicrobial, and anti-cancer activity, the nanoparticles show great promise as a therapeutic agent. Therapeutic use to characterize them help to confirm their effectiveness and new treatment option. Continue research into their mechanism action, by which mechanism they inhibit the anti-cancer or antimicrobial activity. This is an alternative medicine with lesser side effects. These are manuscript under preparation. These are my references. Thank you.
Thank you. Following this, we welcome Madhukar from Deep Field Garden for comparative analysis of quantum enhanced deep learning model using mediation quantum circuit with resonant in dense net 121 efficient net for lung cancer diagnosis.
ये कॉन्फ्रेंस है अभी तो मैंने पोस्ट किया था AIB वीडियो कॉन्फ्रेंस Good afternoon. So today's my topic is a comparative analysis of quantum enhanced deep learning model using variational circuit with ResNet 80, DenseNet 121 and efficient net for lung cancer diagnosis. So aim of this study is to develop a quantum enhanced predictive model that improve early disease detection with accuracy and integrate quantum with the deep learning. So to classify disease risk from genomic and clinical data faster and more accurately. So already we know lung cancer remains one of the most lethal disease worldwide according to global statistics 2020. It accounts for approximately 1.8 million deaths occurs each year, and in a USA, around the 1.6 millions of people died to lung cancer. So this is actually CT scan of lung cancer and normal lung cancer. So here we can see estimated as standard incidence rates in 2020. So and this is another worldwide map of lung cancer. So what is quantum? Quantum is nothing but bits. This is called, what is the difference between bits and qubits? Bit is a single 0 or 1 state, either on and off state, but in a qubit, it will be possible 0 and 1 can be together. So in a, it's called superimposition. So in superimposition and entanglement and a quantum parallelism. Based on these three methods, quantum computing you can create. And so quantum computing uses a specialized technology computer hardware and algorithms that take advantage of quantum mechanics and to solve complex problems simultaneously. In case of a classical computer, one times only you can solve only single problem, but through the quantum entanglement. What is an entanglement? Means if you are here and another person is very far from you, if both are entangled through the quantum, both of person can be think same thing, and whatever you have seen here, so far from your place also you can see it. So based on this method, so next is through this, one is a 0 and another is a 1. So 0 and 1 state can be superimposed, and through this superimposition on an exciting state, this quantum state, 0 and 1 position can be superimposed simultaneously. And here is a, you can see 0 and 1. 0 and 1 both are entangled state. Suppose if you have taken 2 bits, so you can create 4 different types of bits. But for 3 bits, you can make 8 state. So based on how many features you have taken, so can be created complex number, and through this complex number simultaneously entangled and give your result. So based on this method, so data retrieve, so I am a little bit faster. So classical computer you can do with logical gate, so and OR and JOR gate already you know. So this will not work in a quantum computing. In case of quantum computing, one is a target and another is a control. Based on the target and control, it is a similar to XOR, which is called controlled NOT gate, CN gate. So this and another is a CNN gate. And in universal computer based on the CNN method. This is a little bit logical gate, similar to logical bit, and so many gates are possible in a quantum: Hadamard gate, Pauli G gate, Z gate, X gate, Y gate. So I have used Pauli G Z gate. So in this condition, if suppose if you have twice your point in a area, so only you don't know certainty which one will come. Okay, head or tails. Only at the position you have seen it, so only you can see only one position. So similar way, numbers of features is there. So combination of this all the features, personal can be create single result. So on the basis of this Pauli G gate, you can create it. This is a some mathematical equation. So on the basis of the Pauli G and CNN gate, we have made variational gate. On that basis of the variational gate, integrated with the deep learning method and created a model. So 3 different type of model I have integrated: resonate, density, and efficient rate. So for that I have taken a lung cancer data set, a malignant and benign data set, and normal data set. And I have divided this data set 80 to 20% in training set and test set. And after the splitting of data set, going through the image processing. What happened in image processing? You have taken image, so resolution size is a different, but you have to need a fixed size. So I have fixed the size of the images and then converted into the 1D vector, 2D to from 2D to 1D vector in a help of Google Colab platform with a PyTorch interface. So this is a QNN architecture of lung cancer diagnosis. First, I have taken a lung cancer CT scan and go through the ResNet, DenseNet, and EfficientNet and quantum feature encoding, and after parameterized created hybrid feature vector, and then I have go through the CNN model and drop out a average pooling network, created final model, and so on the basis of the on the basis of the parameter like accuracy, confusion matrix, loss, specificity, sensitivity, ROC curve, precision, recall. So get some result. So the combination of parameterized quantum evolution and classical data encoding is successfully demonstrated by the 4 quantum bits. So here is a confusion matrix result, and this is a classification result based on performance. So here we can see DenseNet is a 0.9521, and in terms of train set accuracy and validation set accuracy and specificity, sensitivity in terms of recalls and F1 score, DenseNet showed very better than the all other model. So this is a accuracy chart for the ResNet, DenseNet and efficient net, and this is a loss chart for the ResNet, DenseNet and efficient net. So here is a histogram plot for the specificity and in terms of sensitivity. So here is there is a 2 stages. Stage and first is in a first phase involves resizing the lung cancer images using quantum circuit, and another is another phase we have used CNN architecture to detect lung cancer images. So we have developed quantum enhanced deep learning classification model to detect lung cancer using quantum variational circuit, and the best model quantum enhanced deep learning model showed accuracy value in terms of value validation data set 94%, specificity 96.3.4, specificity, sensitivity, precision and recall. So this research demonstrates how quantum enhanced deep learning can significantly improve disease prediction accuracy in complex biomedical images tasks, and we believe that this hybrid approach opens up a new frontier in medical diagnosis, emerging the precision of quantum computation with a robustness of deep learning. Okay, thank you. These are the references. Thank you. If you have any questions? So I will a little bit faster because of what I do. So if anybody say.
Thank you so much, ma'am, for timely completing your session. Thank you so much. It was a great and learning session. Do you have any question? No. Okay, and Matt is here because you may interact later on as well. So thank you once again, ma'am.
Now I request all the students to proceed for their lunch. Next, I request Anjali Dura for their application.
Yes, ma'am.
Angelika, you can finally proceed with your presentation.
Oh, okay, sure, ma'am.
And you have only 5 minutes to conclude your presentation because we are running short of time, okay?
Okay, for sure, ma'am. Yes, thank you.
बोल दो कुछ में ये बच्चे कह रहे हैं इनकी लंबी सबसे पड़ती है इनकी लंबी सबसे पड़ती है
कौन कह रहा है?
ये दोनों
इन दोनों कह रहे हैं रवि सर से कह रहे हैं? रवि सर सर वो दोनों को नहीं बोलने का टाइम के लिए क्योंकि वो प्रॉब्लम थोड़ी कहते हैं हार्टी के पास स्लाइड शो Angelica.
Angelica, you can start now, and then please do it in slideshow.
Yes, sir. I'm trying to share my screen.
Hi, Angelica. It's fine. You may proceed with your presentation.
So
Okay.
can you
Yes,
can
I
you see my
your-
slides?
Yes. So proceed with your presentation.
Okay, thank you so much, sir. Yeah. Good afternoon, everyone. I am here to present my the topic of my presentation is proteome-wide identification and comprehensive profiling of intrinsic disorder in the liver fluke fasciola gigantica. And fasciolasis is caused by the liver fluke fasciola gigantica as well as fasciola hepatica as a widespread neglected tropical disease which affects millions with significant agricultural and economic losses. Intrinsically disordered proteins lack a fixed 3D structure, providing functional advantages critical for pathogens. Structural flexibility and binding promiscuity rules in immune evasion and host pathogen interaction, which also drives liquid-liquid phase separation. IDPs are well studied and recognized as key virulence factors in viruses, bacteria, as well as unicellular parasites. However, despite the wealth of proteome sequences from multicellular parasitic helminths, studies on IDPs in these organisms is very limited. In this study, we have addressed this gap by conducting the first proteome-wide identification and comprehensive profiling of intrinsic disorder in Fasciola gigantica, and this study highlights the importance of IDPs in the biology of this parasite and provides insights into their potential roles in the parasite's pathogenesis and interaction with the host immune system. These are the methodology that I have used. First, I have retrieved the proteome from UniProt. Then after that, I have carried out composition analysis of the proteins. Then I have predicted the intrinsic disorder using various disorder predictors. After that, I have quantified the disorder and analyzed. Also, I have also carried out CA-CDF plot analysis. Then I have find out the most and least disordered proteins, the top most least and top most disordered proteins, least disordered and most disordered protein. Then I have carried out LLPS prediction using various predictors. Then charge residue distribution, and after that I have compared the IDPs.
Excuse me, Ms. Angelika, I request you to conclude your presentation as we are short on time.
Actually, I have just started. Okay.
You can
sorry, sir.
Then conclude it in 2 minutes.
You can conclude it within 2 minutes.
Okay. And the main conclusion of this study is the Fasciola gigantica proteome is rich in IDPs, and these IDPs are likely to aid the parasite survival and adaptability in the host. And this high disorder provides functional flexibility for dynamic host-parasite interaction, and these insights may help identify novel therapeutic targets in the future, and experimental validation is needed to confirm the function of IDPs across this parasite's life cycle. And where next step, and the next step, of course, is experimental validation to confirm the function across the parasite's life cycle. And thank you. If you have any question.
ठीक है, तो ये भेज देना आपको और ये तो अपने पास ही रखना, ये सर्टिफिकेट डीसी का।
Thank you, Ms. Angelika.
ठीक है, yes, thank you, sir.
Thank you so much.
सर्टिफिकेट हो गया?
Thank you.
Thank you, everyone. तो बोल कुछ
Thank you everyone for the session.
Everyone is requested to
Everyone requested to
For the first presentation.
go for their lunch and reassemble online for poster session.
2 PM फॉर्मेलिटी कर रहा है भाई। ए बंद कर दी वो मीटिंग।
क्या हुआ है? अब मैंने बोल दिया कि
नहीं चल दे तो
मीटिंग चलते ठीक है।
जी मैम।
मेरा फोन देना वहां से तनीश।
Yes. Yes,
Am
you
I
are.
audible, sir?
Yes.
Yeah, sir. Good afternoon, honorable keynote speakers and guest of honors. I am Shefali Sharma, research scholar from MIT University, Rajasthan. Yeah, and first of all, sir, thank you so much for giving me an opportunity to present my paper on this in this conference. So, topic for my paper is startup lead
Excuse me.
sustainability.
Can you
Yes,
please
sir.
presentation mode?
Sorry, sir, pardon.
Can you please present it in presentation mode?
So, presentation. Yes, sir. So this is the, sir, can you see the poster, digital poster for the presentation?
Shefali, you can see the poster, but can you please make it in slideshow?
Sure, sir. Sir, is it visible now?
Shefali, can you please zoom?
Yes, sir.
Can you please zoom in?
Yes.
Shefali, you can continue.
So can I continue? Yeah, thank you, sir. So I am presenting my paper on the topic Startup Lead Sustainability, a study on agri-tech innovation and its role in transforming modern farming. So agri-tech startups are the companies that use technology to solve problems and in agriculture industries. And these startups aim to increase efficiency, reduce waste, and improve sustainability in farming practices. So there are some common practices or common areas wherein agri-tech startups operate. These are precision agriculture, vertical farming and indoor agriculture, agri-biotech and farm management softwares, supply chain and supply chain managements. So agri-tech innovations and agri-tech innovations as we all know that is a new business that's set up and that use technology for moving up value chain of agriculture. So in this paper, it is discussed that how agri-tech startups works in enhancing income, production, and livelihood of farmers as a whole. So in this is a systematic literature paper wherein it discusses that how it works, how it works, which is based on PRISMA, that is preferred reporting items for systematic review and meta-analysis flowcharts. Also there is dependent upon the RStudio software, how it works with bibliometric analysis and maps. So this paper examines the following topics. The main aim or objective of my paper is to comprehend the challenges faced by agriculture sector in India and the study enabling technologies offered by agri-tech startups in India. So there are some challenges as well, which is studied. So the challenges were inefficient supply chain, middlemen and agents, lack of financings, seeds, small and fragmented land holdings as well. So here is the literature review for the paper, which was based on some precision farming and IoT. Precision farming is an agri-tech advancement, including utilization of IoT gadgets to gather real-time information on soil well-being, trim conditions, and natural variables. This all the review of literature was also based on the impact of farmer livelihoods, which was categorized between cost reduction, increased productivity, increasing sustainable farming practices, financial inclusion, better market accesses, improved crop managements. So also, as I told you that this paper was based on Prisma as well as bibliometric analysis of
But you exceeded your time limit.
Yes, sir.
You've exceeded your time limit. You can continue.
Thank you. Thank you, sir. So this was based on Prisma as well as bibliometric analysis also. And these were the bibliometric maps, which was based on co-occurrence network and also articles evolution from 2004 to 24. The methodologies, as I told you, this was Prisma and also our studio softwares. So findings for the paper was that the role of technology in agriculture and farming practices are innovation and revolutionizing agriculture practices while increasing efficiency and also reduce losses. So conclusion for my paper was the study was given a comprehensive investigation on the part of agri-tech sector. New companies are adopting technologies and focusing on sustainability of farmers basically. And looking by the bibliometric information from Scopus, it is found that agri-tech has experienced an ultimate surge in attention and innovation over the past decades. The focus on precision farming, IoT applications, and big data analytics represents leveraging technologies to tackle agriculture challenges. So this was my paper, sir. Thank you for giving me the time.
Thank you, Shikari.
Thank you so much, sir. Thank you.
Next up, we have Yash Sharma. The poster is Impact of Climate Change on Vector-Borne Diseases.
आयुष उनका पोस्टर नहीं आया मेल किस नाम से आना था किसने भेजा है
इन्होंने भेजा होगा।
दीदी को बुलाना है
दीदी
आकांक्षा
दीदी क्या हो गया पता नहीं क्या
मेल नहीं आया
आया कि नहीं आया
डीके सिंह
गया गया अब देखो बता रिफ्रेश हाँ इसे पिन का ओरल टॉक है बाकी सब लगभग उसके बाद नहीं आ रहा क्या ये यार मेरी तरफ से तो चला गया देखो
आया अब
आया ना हाँ करो
एडिटिंग करो नहीं बने इसके बाद फुल स्क्रीन
तब क्यों आ
रहा स्लाइड बना रहा हूँ Listen to along with your PPT. So we'll give you chance again. In the time being, let's continue with the first presentation. Thank you again. We'll start the next presentation.
She is having that. Next we have Ayush Khan. Ayush Khan के present poster इसमें है.
हाँ yes
Next we have Ayush Bal. He'll be presenting on from vaccine to drug target discovery, genome-wide toxin, antitoxin profiling, poisons, Mycobacterium incidus running as the missing link in tuberculosis research.
Good afternoon, sir. I am Ayush Bahal, fifth year PhD scholar under the supervision of Dr. Diksha Tripathi from Central University of Rajasthan. Today, I will be presenting my poster from title From Vaccine to Drug Target Discovery, Genome-Wide Screening of Toxin Antitoxin Profiling Positions, Mycobacterium indicus pranae as the missing link in tuberculosis research. First, we can go through the introduction. Mycobacterium indicus pranae is a non-pathogenic mycobacteria commercially available as a vaccine for leprosy called Immunovac. Owing to its immunomodulatory properties, MIP shows promise as a therapeutic target against tuberculosis and has been a promising target for immunomodulator for COVID-19. TS systems are genetic modules which has two components, a toxin and an antitoxin. And we believe that Mycobacterium indicus pranae is at the intersection of pathogenic and non-pathogenic mycobacterium. Investigating its TM modules may be crucial for understanding the role of TM modules in evolution of virulence attributes in mycobacterium. The aim of this study is comparative survival analysis to stress and antibiotics between Mycobacterium indicus pranae and Mycobacterium smegmatis for establishment of a better study model for tuberculosis research, and genome-wide screening of novel toxin antitoxin modules for exploration of their role in adaptation in Mycobacterium indicus pranae and to use them as a novel drug target. We can move on to results. The first result shows a comparative genomic analysis of virulence factors, drug targets, human homologs, along with the growth rate of Mycobacterium indicus pranae and Mycobacterium smegmatis. It is believed that the virulent Mycobacterium species are very slow growers and the non-virulent Mycobacterium species are very fast growers. So we, by the growth rate, we can conclude that Mycobacterium indicus pranae is in between the both pathogenic and the non-pathogenic species. Second result shows the sensitivity of Mycobacterium indicus pranae and Mycobacterium smegmatis to various abiotic stress conditions. First stress conditions being SDS, nitrosative stress, pH, CuCl2, and temperature. Here we can see that Mycobacterium indicus pranae clearly outperforms Mycobacterium smegmatis. The third result is sensitivity of Mycobacterium indicus pranae and smegmatis to biotic stress conditions. Those include the first line drugs against tuberculosis, isoniazid and rifampicin, and also the second line drugs, gentamicin and levofloxacin, in which Mycobacterium indicus pranae shows better survival. And we can also see the lysozyme stress. The Mycobacterium indicus pranae survives better than the non-pathogenic Mycobacterium smegmatis species. The fourth result shows the quantification of biofilm between the two bacteria, in which Mycobacterium indicus pranae showed better biofilm formation, and the intracellular survival of Mycobacterium indicus pranae in macrophages cell line is also better. And now we move on to the cytokine profiling. Mycobacterium indicus pranae shows elevated expression of both anti-inflammatory and pro-inflammatory cytokines when infected on macrophage THP1 cell lines. The fifth result shows a heat map comparison of percentage identity of stress responsive genes in our control organisms, that is the Mycobacterium tuberculosis S37RB. And we can see that among all the other Mycobacterium species, Mycobacterium indicus pranae shows a better sequence similarity to all the genes that are responsible for some kind of virulence that includes TS systems, chaperones, trigger factors, and other genes. The next result shows now we have established Mycobacterium indicus pranae as a better study model for tuberculosis research. Then we went on to the second objective of the study, that is the genome-wide screening of TM modules in Mycobacterium indicus pranae. Now, I think there is some problem with the poster. My result has shifted towards the other side. The result 10 and 11 shows the PSI blast results of all the putative toxin antitoxin systems in Mycobacterium indicus pranae with other microorganisms in question. Those have very found role in toxin antitoxin systems that include Mycobacterium tuberculosis, Vibrio cholerae, E. coli. And now we can move on to the seventh result. It is the heat map showing all the gene of all the putative toxin antitoxin modules in Mycobacterium indicus pranae with all the Mycobacterium species that are present. And now when we have all the we have come to the conclusion that Mycobacterium indicus pranae has many TM modules, and it has an intermediate number of TM modules that is between the pathogenic Mycobacterium tuberculosis and the non-pathogenic Mycobacterium smegmatis. So we narrowed down our research to 3 TM modules, and we performed a QRT PCR to see that if these genes actually express themselves in stress conditions, and we can see a clear expression of my TM modules in all the stress conditions that have been explored before. Now, we believe that TA modules are responsible for persistence in bacteria. So if somehow we can stop the interaction between the toxin and antitoxin module, we have designed a peptide inhibitor so that it can bind to the antitoxin, and toxin activates itself, and it is a very good drug target. And now I can conclude my presentation. MIP represents an evolutionary bridge between Mycobacterium smegmatis and Mycobacterium tuberculosis. And this study was significant because this is the first time we have genome-wide screening of TM modules in Mycobacterium indicus pranae, and it establishes Mycobacterium indicus pranae as a better study model for tuberculosis research. I would like these part of my research has been already published into for 2 papers, and the third part is coming on. I would like to thank Dr. Diksha Tripathi and Central University of Rajasthan, and Professor Rakesh Bhatnagar from GNU for providing all the strains necessary for this experiment. Thank you.
Next, I invite Ajay Prajapati on post titled Evaluation of Radiation Exposure in Pediatric Patients for Minimizing Radiation-Induced Risk in a Tertiary Care Hospital. He's not here with us, so I'll move on to next.
Next up, we have Nikita Kumari Pandey from Vanestali Vidyapeeth. Tools and technologies for rapid, accurate diagnosis of plants and animal diseases.
Okay, thank you. Good afternoon, sir. I am Nikita Kumari Pandey from Department of Biosciences and Biotechnology, Vanestali Vidyapeeth. और ये जो मेरा पोस्टर है वो एक रिव्यू आर्टिकल है कुछ ऐसे टूल्स एंड टेक्नोलॉजीज का जो हम यूज करते हैं करंटली प्लांट के और एनिमल के डिजीज के डायग्नोसिस में। सो हम जैसे कि जानते हैं कि जो प्लांट और एनिमल डिजीज है वो बेसिकली बहुत बड़ा ग्लोबल थ्रेट बन चुका है फूड सिक्योरिटी के मैटर में। सो हमारे पास बहुत सारे जो ट्रेडिशनल मेथड्स है वो हम यूज कर सकते हैं, बट वो उनका सबसे बड़ा ड्रॉबैक ये है कि वो बहुत ज़्यादा टाइम ले लेते हैं। दे कैन टेक फ्रॉम डेज टू मंथ्स फॉर रिजल्ट्स। Therefore, the modern tools and technologies come to the market. And first we have the molecular diagnostic tools in which we have the PCR, LAMP and CRISPR-based technologies. Then we have immunological technologies in which we have ELISA and LFDs. Then we have some biosensors and nanotechnologies in which we have some graphene-based technologies, gold-based technology, biosensors, sorry, optical biosensors. Then we have imaging and remote sensing technologies in which we have hyperspectral imaging and thermal cameras and drones equipped with AI. सबसे बड़ा गेम चेंजर यहाँ पे जो है वो AI का है। AI बेसिकली बहुत AI का यूज करके लोग एक ऐप बनाते हैं, मतलब ऐप डेवलप करते हैं जिसके थ्रू जैसे कि उसका एग्जांपल है प्लांटिक्स एंड लीफ डॉक्टर। इसमें जो फार्मर्स है वो अपने प्लांट्स के इमेजेस अपलोड करके और उसको कंपेयर कर सकते हैं विद द हेल्दी प्लांट्स so that they can confirm if their plant is infected or not. Then we have some network platforms like One Health and integrational diagnostics platforms in which global farmers can interact with each other and share their problems and conclusions in for the disease and their spreadings. I will conclude my poster with this that ये जो टूल्स एंड टेक्नोलॉजीज हैं इनका यूज करके हम डेवलप कर सकते हैं कुछ ऐसे प्लेटफॉर्म्स या फिर ऐसे एप्स जो कि डिटेक्ट कर सकते हैं प्लांट में जो डिजीज है उसके इमर्जिंग के पहले उनका डिटेक्शन कर सकते हैं और उनके प्रिवेंशन के आइडियाज दे सकते हैं थैंक यू सो मच
Next, we have Mahika from Banasari Vidyapeeth on topic transgenic approaches for enhancing disease resistance in crops, molecular strategies, and field applications.
जूम नहीं होता क्या अब जूम भी कर सकते हैं ना मूव कर सकते हैं ठीक है
Good afternoon, everyone. I'm Mahika and I'm Umal Ranawat from Department of Bioscience and Biotechnology from Vanasthali Vidyapeeth. I'm here to present our poster on transgenic approaches for enhancing disease resistance in crops. Facing high global population, crop protection is paramount for food security. Transgenic approaches offer a sustainable pathway by introducing specific traits for enhanced resistance and yield. Central to this biotechnology is Agrobacterium tumefacium, a great genetic engineering tool, natural genetic engineering tool, which helps to transfer DNA into the plant genomes. Now we will look into the following three molecular strategies. Number one, overexpression of pathogenous related proteins. Pathogenous related proteins are the proteins produced as a part of the plant's home defense response system against the pathogens. Their overexpression means genetically engineering a plant to produce these proteins in a larger quantity than they normally do. Here in our poster, we have attached the systematic pathway, like how these plants are genetically engineered to increase their production. The next method is expression of antimicrobial peptides. Antimicrobial peptides are small cationic bioactive peptides present in plant system which possesses antimicrobial activity against several pathogens. Now the third approach is the pathogen-derived resistance. This concept is based on the idea that a plant can use a pathogen's own weapon against it that disrupts its infection cycle. How Agrobacterium enables a PDR. Pathogen gene is selected, gene is inserted into the vector. Agrobacterium carrying the vector infected the plant genome. The tDNA is transferred to the plant genome and the transgenic plant cells are regenerated. Now, let us look into a case study on late blight resistant potato via RPI genes and Agrobacterium tumefacium. Late blight is a disease caused in potatoes via a fungus-like microorganism called Phytophthora infestans. RPI genes provide us resistance against this disease. These RPI genes are found in the wild variety of potatoes. We can transfer these genes into the potato variety through Agrobacterium mediated transformation. Now we have two field applications. First, the rainbow and Sana papaya. The other is the late blight resistant potato, which we already discussed. Papaya is highly susceptible to papaya ring spot virus, which leads to significant yield loss. In 1990s,
Scientists developed transgenic papaya varieties through expression of PRSV code protein via agrobacterium mediated transformation. This successfully controlled the PRSV epidemic and allowed farmers to resume cultivation and previously devastated areas.
Now, we would like to conclude by saying that agrobacterium mediated transformation remains a major source of genetic engineering tool, and through technologies like overexpression of PR proteins, expression of antimicrobial peptides, and PDR technology, we can help reduce these pathogen effects. Thank you.
Next up, we have Sumaran Chaubat from LIMS University on the topic a review article on new and re-emerging animal diseases, early response and risk management. Since he is not there, next up we have Nikita from Sanskaran University on topic integrative approaches in IPM harnessing weed-based bioinsecticides and in silico tools for sustainable rice health management. She will be joining us online.
Nikita? Am I audible to you, Nikita? Nikita, you can proceed with your poster. Yeah. Next.
Next, we have Sharma and Sanjita Singh from Sanskaran University, nanotechnology enabled precision weed and disease management, smart delivery systems, and sensor-based strategies for sustainable soil agriculture.
Shama, am I audible to you?
Yes, ma'am.
Yeah, you can present your poster. You have 5 minutes.
Okay, ma'am. Good afternoon, everyone. My name is Sama and I am from the Department of Botany, Sanskaram University. Today I am here to present my work on nanotechnology-enabled precise weed and disease management, smart delivery system, and sensor-based strategies for sustainable. Hello, yes, sir.
The poster is not presenting. Can you present your poster? Can you share your poster?
Sir, is now my screen is visible?
Yes.
Okay, thank you, sir. Oh, sorry. Now my screen is visible now, sir?
Yes.
Okay, so today I want to talk about a big problem that farmer faces weed and disease in crop like tomato, potato, eggplants and chilies. These crops are very important for our food and economy, but weeds and disease destroy a lot of them every year. Weed like Amaranthus, Chenopodium, Solanum nigrum, many grow very faster in field and compete for nutrition, water, and sunlight and cause disease like late blight, bacterium wilt, early blight, powdery mildew, and root knots. Because of this problem, every farmers can lose 30 to 40% of their crops, which means millions of rupees and loss every year. To fight these problems, farmers often use some chemical herbicides and pesticides, which is not good for soil, not good for our health, and also do some environment pollution. So here is also a solution which could replace chemical herbicides or bioherbicides, natural weed killer made from weed like lantana and parthenium. By these weeds extract, we make natural compound that stop weed growth without harming the crop or the soil. When bioherbicides are combined with nanotechnology, they become even more powerful, long-lasting, and eco-friendly. As we know, nanotechnology is nowadays uh, very known, and in farming, we can use this for deliver crop medicine precisely, reduce chemical use, and protected the environment. Nowadays, as we all use smartphone, widely use smartphone, these is there is some application already available on Play Store like Agrio, Plantix, and Tournament. There is so many more application also have. So farmer has become even smarter with AI, drones, sensor, with for their crops. They can use this oil for their crops. Drones scan the field to detect weed patches or disease plants, and sensor measure water, temperature, nutrition. Then AI analyze this data and decide exactly where and how much nano spray to use. This method, known as precise farming, helping farmers save money, save time, save resources while protecting the environment. For these all tools, farmer will use their phone, because all we can use AI tools and drone sensor. We can detect all the disease by their phone. So last now we can conclude this with nanotechnology and bioherbicides and AI are helping farmers protect their crops in smarter and safer ways. This method reduce chemical use, keep the soil healthy, and increase crop yield. Thank you. Is there any question? Hello?
Thank you.
Hello?
Thank you, Nikita.
No, sir, it's me, Sama.
Thank you so much.
Your time is up. Okay. You can log out from this session.
Okay, now can I stop sharing my screen?
Yeah, you can log out from this session.
Okay, thank you.
Next, Ankit Parashar. Next up, we have Ankit Parashar from RIMS University on topic environmental impacts on migratory birds in Rajasthan, patterns, challenges, and conservation strategies. Since he is not here, next up we move to the S. Sarvendran and Ayushi Tiwari from Amity with topic an in silico study on inhibitory potential of phytochemicals against an all-Indian serotype of dengue targeted virus, NS5-RDR.
Good afternoon. I am Ayushi Tiwari from BTech Bioinformatics along with my batchmate S. Harvendran, and we are here to present our poster on the topic an in silico study on inhibitory
Potential.
inhibitory potential of herbal components against dengue virus. So basically, dengue virus is a mosquito-borne flavivirus that present a significant global health risk, especially in the tropical region with no significant antiviral treatment. So here, what we did is we took out RNA-dependent RNA polymerase, part of NS5 protein, and which is highly conserved across the all the four stereotypes. And here is the diagram of all the stereotypes and serves as a prime candidate for the drug targeting. The study basically focuses on the utilizing Indian stereotypes and maximizing the regional relevance, aiming the discovery and promising pharmacochemical and that can act as inhibitor against DNA RDP.
Now I'll explain the methods which were taken to perform this paper. A consensus sequence of RDRP catalytical domain, which was derived from Indian dengue viruses across four serotypes were selected, and 125 sequences of these four serotypes were consensus into a single sequence for each serotype. As you can see the structures over here, which was created by AlphaFold using ab initio structure modeling and validated by Project using Ramachandran plot and other parametry. And we followed it by docking studies using LibDoc module in Discovery Studio using phytochemicals which were chosen from literature of different papers, which was proven to be working on a single serotype, which is not worked on multiple serotypes at the similar time. Because in dengue virus we cannot predict the serotype which we have within our body, so it is important to affect all the four serotypes for the drug to be useful. So this is the main part of the paper, and the phytochemicals which showed a good affinity towards the proteins were Chartacon family, which were evaluated with a good docking score. As you can see, the results of this docking score. The Chartacon family A1, A2, A3, A4, and B, C, D, E, F were showed a significantly good score on all the serotypes. The threshold of this docking score is 60, but we got more than double of it. And these were the methods that were used for this selection of this ligands and their screening. We selected this ligand, and these ligands were screened using pharmacodynamics and pharmacokinetics for the drug likeliness using Swiss ADME and PREDMET. The lead candidates were prioritized for high gastrointestinal absorption and compliance with Lipinski rule, other drug likeliness, and the toxicity prediction was done using ProTox, which shows us the respiratory and carcinogenic and other toxicity thing. And PREDMET servers is an in silico tool for animal model based toxicity prediction. We ran it on rodent and like mice and other animal models. For it came out to be non-carcinogenic and non-mutagenic. And I will pass on the mic to Aishi for discussion and conclusion.
So basically, Chartacyon B and Chartacyon A1, A2, A3 were based on predicted high gastrointestinal absorption, and they followed Lipinski rule of 5 as well as ADMET properties. So it shows basically that it has a drug-like property. Also, when checked for toxicity, it gave positive results. And in silico study successfully identifies the 5 Chartacyons, that is B and A1, A2, A3 and A4 were derivatives and potent bioavailable candidate inhibitors for dengue RDRP. This component shows strong binding, favorable pharmacokinetic and excellent safety, warranting future work involving molecular dynamics and simulations. Thank you.
Thank you.
Next.
Thank you. Next, we have Pradeep Kumar from Amity School of Applied Sciences with quantifying disease burden and life expectancy loss from biomass emission in rural North India.
सुन रहे हो क्या? हाँ हाँ सुन रही हूँ. हाँ बोलो बोलो इनसे बात करो.
Final thesis?
Hello, I'm audible. Hello, I'm audible.
किस नाम से?
Presentation.
Nikita, you have to wait for some time.
Okay, ma'am. Okay.
ये
ऐसे निकलता है आपको बिल्कुल डाउनलोड कर लिया
ये क्या?
हाँ ये agricultural waste new moves.
ऐसा कर इनके बाद इनको इनके बाद
particulate matter as well as gases, which causes health impacts, respiratory as well as cardiovascular residents.
अभी मैंने मीटिंग पे शेयर किया.
This study fills gap by quantifying the disease burden of life expectancy loss attributed to biomass conditions in two rural districts, Jhunjhunu in Rajasthan and Mehrgarh in Haryana. They conducted a cross-sectional survey of 1,000 households, 500 from each district.
मैं एक बार थारी कमरे में जाऊँ यार और तुझे
This survey on economic ventilation and fuel type residents were using.
हेलो हाँ क्षमा.
As well as self-reported clinical verified illnesses like asthma, hypertension, diabetes, and eye irritation. Health outcomes were categorized into respiratory, cardiovascular.
I'm audible.
Yes, you're audible.
Okay, thank you.
Already 5 minutes.
My screen is visible.
Yes, it's visible.
Okay, thank you. Good afternoon, everyone. My name is Nikita. I am a research scholar at Sanskaram University. Today, I am representing a poster on the topic Integrative Approaches in IPM, Harnessing Weed-Based Bioinsecticides and In Silico Tools, Sustainable Rice Health Management. Today we are talking about the disease that is caused by Nila Parvata lugans on rice, Oryza sativa. Oryza sativa is a staple or edible crop for more than 50% of global population, and almost 3.9 billion people feed on Oryza sativa. Brown plant hopper, that is Nila Parvata lugans, is a destructive pest that cause a major yield losses in rice, and we need one health-based solution for the pest management. There are huge challenges. First is chemical insecticides, and second is biotic stresses. Some limitations of chemical insecticides are pest develop resistance with repeated use and harmful. Yes, sir. Harmful, harmful harmful effects on soil health, biodiversity, and beneficially insects. Its high cost also makes and dependency for farmers. And second is biotic stresses, that is virus vector that transmit RRSV and RBSV that cause rice yield losses. Second is hopper burn. It feed on the phloem sap of rice and disrupt photosynthesis and kill the plants. Some weeds can be used as a biosource for bioinsecticides that may be Lantana camara, Parthenium hysteropus, or Chromella odorata that contain bioactive compounds that are terpenoids, alkaloids, flavonoids, phenolic, and growth factors that cause that can stop the growth of Nila Parvata lugans. Its effect is oviposition and repellency mechanism that cause antioxidant growth regulator, enzyme inhibition, neurotoxic effects that are harmful for these insects, these pests. Some integrated in silico or in vitro approaches can be used to control the growth or Nila Parvata lugans. In silico methods are molecular docking, ADMET prediction, molecular dynamics stimulations. In vitro bioassays are used, and phytochemical profiling can be used to stop the growth of Nila Parvata lugans. Sustainable IPM and one health perspective. Weed crude extract could play a crucial role in reducing chemical pesticide uses, improve sustainable agriculture. Weed-based bioinsecticides represent a disruptive innovation for sustainable health management. It will be beneficial for plants, animals, and humans. In plants, it reduces pest resistance and yield loss. In animals, it minimizes the feed contamination. In humans, lower pesticide residues in food. Environment, biodiversity conservation can be saved through it. Conclusion for the poster is eco-friendly alternative. Weed-derived bioinsecticides offer sustainable pest control for rice. Integrated approach liking phytochemical and computational experimental tools enhance the discovery or integrated pest management approaches for the sustainable rice health solution. Yes, ma'am.
Nikita, your
Yes,
time
ma'am.
is up. You can conclude it in 1 minute. Your time
Okay, ma'am. So the integrated pest management is a sustainable way for the rice health management, and the use of weeds as a bioinsecticides are eco-friendly alternative, and weed-derived bioinsecticides that offer sustainable pest control for rice. Integrated approaches or in silico method, in vitro approaches may play a role to solve the problem that is caused by the Nila Parvata lugens against the rice. It causes disease that transmits the virus vector in animals or other organisms that can be saved by using of these bioinsecticides. Thank you.
Thank you, Nikita. But I would like to invite Atul Kumar Yadav, APS Amity University, Rajasthan, on topic Startup Clean Sustainability, a study on technological and commercial barriers to global biosecurity. Since he's not with us, I would like to invite Prashant Sharma, AIB Amity University, Rajasthan, exploring the impacts of molecular interactions of natural ligands with gelatinosis from Entococcus facilacis by in silico approach.
Nikita, in the meanwhile, you can stop sharing the screen.
Anish. Rudra.
Prashant, am I audible to you?
Prashant Sharma, you can start.
Share your screen.
Your screen is not visible.
Prashant, could you please share your screen? It's not visible over here.
Nandini Soni.
Prashant is not responding. Next, I would like to invite Nandini Soni, AIB Amity University, Rajasthan, on topic linking water quality and carbon footprint, a case study of university lake and local aquatic systems.
Since Nandini Soni is not present.
He's not there.
Since Nandini Soni is not present, next I would like to invite R.N. Paste, Rohan Kumar, and Nidhi Jain on topic comprehensive human health prediction.
Unmute है दीदी. Mute कर. आपने मेल किया था?
Actually, screen पे ही है.
Screen पे. Yes, ma'am.
4.5 minutes का strict time आ रहा है. आपको मैं conclude करनी है. Yeah, 4 minutes. 4.5 minutes. Yeah. Hello everyone. My name is Arvind Paste, and I am here with my team, Nidhi Jain and Rohan Kumar. We are from MIT Institute of Biotechnology and from MIT University. We have proposed an idea, and based on that, we have made a poster on human health prediction. This is an hypothetical app, and we will speak about it. Did you know that chronic diseases cause about 71% of global deaths, yet most healthcare systems only respond after illness strikes? What if we could predict health risks years in advance and prevent diseases before they develop? Our human health prediction platform connects the latest AI technology with collective data and multiple sources. It connects to specialized biological databases such as UniProt for proteomics, ClinVR for genetic variants, pharma and drug genome interaction, environmental database for pollution and climatic factors. Our app collects and processes the biological profile of each individual, which includes clinical biomarkers, then compared against detailed profile of pathogens and detailed agents. To analyze the individual susceptibility and chances of infection or diseases, using advanced AI algorithm, the app calculates risk predictions via visual charts of various health threats. The app also provides personalized prevention and lifestyle modifications to reduce the risk of and enhances immunity. Current health prediction tools often lack data integration and real-time updates and detailed personal information, which limits accuracy. Privacy concerns and biases also reduces trust in this system. Our app solves these challenges by continuously updating predictions and collaborating with hospitals and lab researchers, enhancing the accuracy and giving clinical validation. We prioritize user privacy via powerful encryption, local data processing aligned with regulatory standards. This makes health prediction more reliable, personalized, and trustworthy. By combining cutting-edge artificial intelligence, rich databases, clinical validations, and user privacy, our platform shifts healthcare towards proactive prevention. This empowers individual and medical professionals to make informed decisions for better health outcomes while reducing the burden of chronic disease globally. Thank you and have a nice day.
Thank you for your input and congratulations to all the partners for their outstanding posters and continued contributions. This concludes.
You can ask if anyone is remaining and still waiting who could not present. We can take them.
Is anybody remaining? They can now start their posters. Sean Sharma, are you there?
Please mute yourself.
Sean Sharma, unmute yourself. Prashant Sharma, are you there?
Who's this?
Prashant, there is still timer of 5 minutes. You have to complete your poster in 5 minutes. Prashant, you are not audible.
Audible.
Hello everyone, am I audible?
Yes. Prashant, there is strict timer of 5 minutes. You have to conclude your poster presentation in 5
Hello.
minutes.
Am I audible?
Yes, you are audible.
UPI है देश के प्रमुख प्रेजेंट
Hello.
Prashant, you have to conclude.
Hello. Am I audible now?
Yes, you are audible.
Hello.
Prashant, you're audible. Please continue with your presentation.
Okay. No, I'm not able to listen to you. Your mic is off, I think.
You speak to देश दीपक क्या ये Since you're not audible. अरे भाई क्या हो गई हाँ क्या लिखना दीदी
We will mark your attendance.
Attendance has been marked.
ठीक है बस
हाँ.
बोल युक्ति का नाम
Dr. Dean, is evaluated. We have seen your posted.
ठीक है तू बोल दे मैं लिख लूँ
I'm not able to listen to you, but I think I am audible. So if any query, please drop a message because I don't know why you are not audible for me. So I think I should start presenting.
Next up, we have Yuki, who will be presenting to Desh Deepak sir.
प्रशांत आ गया प्रशांत आ गया
Prashant, can you please speak in control if you're
I'm
audible?
not able to listen to you, but
You're audible now.
I am audible, so I will start this presentation. If there are any query, please ask in chat box because there is some technical issue. I am not able to listen to you.
प्रशांत अब आपकी आवाज प्रशांत आवाज आ रही है आपकी
My topic, good afternoon everyone. I am Prashant Sharma, PhD scholar under Dr. Sanket Kaushik from Amity University, Rajasthan. My topic is exploring the impact of the molecular E. faecalis by in silico approach. My research motivation is because E. faecalis is most common. E. faecalis most common etiological agent responsible for hospital-acquired infection, a nightmare for the health system. E. faecalis rapidly becoming multi-drug resistant, owing to its ability for forming biofilm. Hence, it's crucial for treatment strategies against E. faecalis. Non-selective use of antibiotics has increasing terminally due to the such incidence to occurrence. MDR bacteria is growing fast. E. faecalis is one of the most responsible for disease, and it's also come escape pathogens, and the normal habitats it's the gastrointestinal tract of human and animal. It is also responsible for UTI, infection and inflammation to GI tract infection and pelvic organs, wounds, and endocarditis. Gelatinase E or gel is a crucial virulence factor, zinc-dependent metalloproteinase of the AMPO family that degrade host macromolecules and promotes biofilm formation and dissemination. Objective of this studies characterize E. faecalis structurally and identify potent low cytotoxicity natural inhibitors to combat MDR. For this, I use three major methods: protein modeling and validation, uh, interactome analysis, and molecular docking. For protein molecular validations, and I use AlphaFold 3 for modeling, and for the validation of the structure, I use ARAT and Ramachandran plot. With this, I found that this protein is a globular protein with 16 alpha helix and 12 hairpin loops, and they consist active size motif HEMTH. The location of that is 327 to 331 amino acid location. Then I go for interactome analysis with the help of string version 12 to detect the protein interaction network. We found there are two key partners with higher confidence, more than 83%, is FSRA responsible for regulation protein, and EF1817 that is protein serine protease. FSR positive regulator of jelly expression, crucial for quorum sensing system and biofilm formation. And serine protease play a major role in another valence factor. The key takeaway was that jelly is a pivotal node in the quorum sensing and biofilm regulatory network. After this, we go for molecular docking with Autodoc Vena. The target site was HEMTH motif. The ligand with three ligand used, latinin, genquanin, and taxifolin, were tested against standard quercetin. After blind docking, we found three suitable chemical compounds which we use from after review studies. We found this ligand, and after blind docking, we found this suitable. So we go with all